1, 2, 3, testing. Thank you all for joining us. We've just currently landed on the seabed for dive number 244.
That's a new waypoint for you.
At the moment.
From there, then you'll be able to do trash traction.
Yeah. I mean, it's like the sun says, it's like waypoint 26 will start.
1, 2. Wonder why I could hear you. It's weird. Okay, so we've landed on the seabed for dive 244. We're currently heading towards Alvin Spire. That's not mine. This will be a picturesque vent.
Some bacteria.
En route, we may see some of the junk that you've seen. If not, at one point today, we're going to be transiting along what is a
4037. All right, let's continue towards Alvin Spire.
Give you a bearing. Yeah. It's funny how the markers don't appear on my chart, but they waypoints do.
135, 140 meter, 305.
305, right?
Is that paradepth or
We should be on INS depth.
INS?
We are on Paro depth.
Are we?
Oh, it's para. Thank you, Jesus. 'Cause
Oh, INS. Are you feeling the tight right now?
A little
is
bit.
it?
Oh, that's right.
Okay.
It's on INS.
All right.
It's 70, so it's delta 50 the whole time. Has it been feeling like that the whole time?
It was pulling a bit, but.
And a good morning to Jackie Pearson, or good afternoon in the UK,
Funny difference.
Well, you were
in a sunny, windy Southampton. And hello to Jamie. Thanks for joining, the usual. At the moment we're transiting towards Alvin Spire, a truly spectacular hydrothermal vent. This was dived by the the human-occupied vehicle Alvin, as part of one of its 5,000 dive. Alvin's been around since the 1960s.
Yeah.
Unlike
there's a
Sebastian,
sea
which has been
spider.
around for 3
See
years,
that?
and
We haven't
has
seen one of those
completed
here.
244 dives.
Ooh, fish. Good.
Now, if any of you have any questions, feel free. You can ask them on our stream. Do our best to answer them.
So it shouldn't have been on this track towards Alvin Spire.
Well, it was a good thing that we are because we found something interesting.
There's a, what's this thing here?
Jackie says hi.
Hi, Jackie.
Good morning
That is the
from
longest
James as well,
clam
Jackie.
trail in the history of the world. Yeah, there's the clam right there.
So at the moment, not a lot to see. That's a clam trail. We're headed towards Alvin Spire. How far away is it there?
I wonder how long it took that clam to go that far. Actually,
We
there
are
it is.
130 meters away from Alvin Spire, slowly making our way across the seabed.
It's a really long way.
These hydrothermal vents are really oases in the deep ocean. Between them, there's very little. You see the occasional clam track. We've seen a lot of octopus at this location.
I'll let you know when they join us.
Okay.
But also, if you want to give me a warning, I told them we give you a warning, they might just let us start.
Yeah.
So-
Okay. Yeah, we should be there in about 10 minutes. How far is it?
Still got a way to
What's
go.
that? Over to your right there. There's a fish.
Yeah.
Slowly catching up.
Oh, okay.
So in the control room, we've got Professor Joy, who's giving directions throughout the dive. Next to her, we've got James, who's currently piloting Sebastian. And then next to him, we've got Adam. And then myself, Leighton, who's managing the live feed and the data coming from remotely operated vehicles, Sebastian. We're currently 2,034 meters below the Gulf of California. This is the final dive of expedition FK 190211. During this expedition, we've completed 15 dives.
Here's a big fish.
There's a big fish coming into view.
Oh,
ROV
there's a
SuBastian
Shawnax.
is
We haven't
remotely
seen those
operated.
yet.
It's currently 2,000 meters below us, being controlled by our pilot and co-pilot through several kilometers of steel armored umbilical. These images are coming to you from our satellite feed on research vessel Falco, operated by Schmidt Ocean
Oh, no, I've
Institute.
pressed that.
Let's use that now.
Yeah.
Remember we had the
Skid, yeah.
During the night, Falco was engaged in mapping operations as well as water sampling operations around these hydrothermal vents. We were using our CTD
This just
rosette,
looks like
which is a large
Rainbow
device
from
that has
Mexico.
24 bottles
Yeah.
on it. We lower it down to different depths, and then we tell about the bottles to close. We can trap water samples at various depths. The water is then brought back on board and studied by our scientists. During this expedition, we've seen some absolutely amazing vents. There's been a lot of different gases coming out of these vents, as well as oil. Those who joined us on yesterday's dive would have seen these amazing globules of oil, as well as some pretty cool hydrate. Yesterday was spectacular. Hopefully today we're going to get some nice images. We should be arriving at Alvin Spire in roughly 10 minutes. So sit back, enjoy the slow, sedate speed with which we're transiting over the seabed. I'm told to read the dive plan. Okay, oh, we'll read it out live on air, shall we? Well, it might not while we're cruising across. So, today's dive is 243. It's the 10th of March here on board Falco, and it's 9:05 in the morning. Our launch coordinates. First of all, we dropped our lander. This is basically an elevator for equipment to and from the seabed. There's already some of our experiments from our previous visit here on the seabed, and at the end of today's dive we'll go and collect those experiments, put them back on our elevator or lander as we call it, and send them back to the surface. Then we launched SuBastian. Took about an hour to descend to the seabed, and the objectives of this dive are to return to Falco's fountain, truly spectacular event that we found the other day on one of our listeners or watchers online named, and there we'll collect some fluid samples from the vent itself, and then we'll head off to Big Pagoda and look around for other pagodas. All the vents around here have the name Pagoda in them. So far this cruise we've explored a few others. This area was previously mapped by the Autonomous Underwater Vehicle Sentry, operated by Woods Hole Oceanographic Institute, and it identified a number of lumps and bumps. During an expedition in November, a team visited some of these features, including Alvin Spire, but there was still a number left over to visit. During this expedition, we've gone to a number of features that were previously unexplored. This included Falco's Fountain, Big Pagoda, Holy Cow Vent, as named by our principal scientist for the reaction that she had when she saw it, Red Dragon Pavilion, which was named because it was St. David's Day, Welsh national holiday, or Welsh national day, I should say. What other ones have we found, gents? Garbage Vent. Garbage Vent so called because of the utterly depressing mound of garbage all around it. So we've found plenty on this expedition, and there's been a lot of surprises. Rebecca's Roost, I think, was found, I'm not sure in November, but in 2008. Maybe Professor Joy
Yep.
can elaborate on previous work here.
2008. This area was described in 2008 and 2009. It was visited before that by geochemists and microbiologists to study, the interesting type of fluid seepage that happens here, and to work on the beautiful Beggiatoa mats. The mats have absolutely received the most attention of any aspect of the system. When the area of Big Pagoda was visited in 2008, it was about a 2-meter high chimney that was not anything approaching the magnificent structure that it is now, and these large spires seem to be a recent phenomenon here. So that's pretty exciting to be able to document and share that with everybody, and we'll be pulling up to Alvin Spire here in just a few minutes.
Okay, Jackie Pearson from the National Oceanography Centre in Southampton is asking, what has the biggest highlight of this expedition been?
Oh, I think it's got to be the mirror flanges at Big Pagoda. I don't know. The hydrates were pretty cool too, but those mirror flanges are just surreal. It's I don't know. walking around in Narnia or flying around in Narnia or something. I mean, it's just an amazing thing to see. And finding it once on Big Pagoda was pretty special. And then we found, 4 other features that have, similar characteristics. And in all likelihood, there are more of them out here, and we just haven't stumbled upon them yet. But we hope to find another one today to share with you all on the live feed.
Yep. And for
What do
now,
you think, Leighton? What was your highlight?
Wow.
Mirror flanges?
I agree. Some of the spectacular mirror flanges that we've seen, this underwater mirrors, that these are unlike any other vents that's seen. Most of the ones we see are just like chimneys, but these are just like, I don't know the best way of describing it, plates and plates stacked on top of each other with all these like reflective mirrors underneath. They're absolutely, truly spectacular. Yeah, I agree there. There's been some depressing sights. I mean, the garbage we've been seeing is just a reminder of the effect we're having on the planet, but in between that, there's these lush oases of life that is just amazing.
All right, we're coming up on the spire. This is a really impressive thing to see. We're going to start at the bottom and fly up. Leighton.
Okay,
Go ahead.
when we first came to some of these locations, especially around the base of Alvin Spire, the other day we were truly disheartened to find what is just masses and masses of rubbish just littering the seabed. So as we progress through today's dive, you'll probably see some locations, and there might be one on the left here, where garbage has been trapped. I'm not sure about this location. Oh, but we are truly at the base of Alvin Spire.
Yeah, here we go. So there is garbage here.
Yeah, there's some garbage trapped to the left there.
Nothing?
Are they the coals we're looking for?
If we cruise around and find the sponge rock, there's garbage there too.
Oh, there's an octopus right in the middle of it all.
There's garbage.
Yeah, there's garbage.
Okay, so we're just going to show you some of the garbage that we found. This just represents a small amount. Further over to our southwest, which we'll probably go and look at on the way to Falco's fountain, is just fields and fields of rubbish and garbage that has ended up at this location. So what we're talking about here is some of the most beautiful vents, and you'll see for yourself shortly, that can be found on this planet. But interspersed around this area, an area that no one's gone and seen before, is huge amounts of plastic and rubbish that has been ended up going into the ocean and ended up at this location. And it is truly disheartening to see what is previously unexplored areas just littered with rubbish. And it really hits home at the impact that plastics are having in the deep ocean. A lot of people don't get the chance to come and see these places, so out of sight, out of mind. But here we are streaming these 4K videos to you, showing you the some of the rubbish that you can find on the seabed. But for now, I think we're going to go and look at Alvin Spire. And later in the dive, we'll be transiting along the trench of trash, as we call it, towards Falco's fountain.
To the left or the right, which is the best?
Let's motor straight up. We're going to start at the base here. There's a sponge garden at the base of the structure. The sponges are occupying the sediment where there's no hydrothermal flow, so the water's fairly cold and innocuous background material, but they still benefit from the hydrothermal flow because material settles down from the overlying water. If you watch carefully in nooks and crannies in the rock, you'll see an octopus. There's one right in front of us now that's hiding back here. You can just see. Can you hold for 1 second? There's an octopus right in here. If I can get him to show him or herself. We are moving, so I cannot. Do you see where I'm going? Right here is the octopus. It's like right in here. It doesn't matter. We can keep going up. There's a nice anemone, sponges, and as we go up the face, you'll see the fauna shift from cold water filter feeding fauna to more high temperature loving fauna, microbial mats, riftia tube worms, anemones, and a whole slew of invertebrates. But I think you'll be surprised by the magnificent colorations of these organisms. The pigments that they use to generate their energy to make their living are comprised of metal, trace metals, and these trace metals are often colored, hence life has color. So we're coming up the backside of the spire, and here's more sponges. We're going to continue. You can see squat lobster crawling up the side of the rock, and some hydrozoans. These are Cnidaria. There are a lot of cracks and fissures on Alvin Spire. She's a pretty vaulted rock, and you can see as we continue to move upwards, there's going to be a very large crack. I think that'll be on the left, if that sort of propagates across the feature. And when we get into the main flow field, that giant fault has extensive flow through it. So here's the fault I was speaking of a second ago, right here. And as we sort of fly up that fault, you'll see that at the end of it, there's a good deal of flow and discharge. So here's the fault. So we're on the opposite side. Yeah, the mirror flanges are on the other side. So this is a pretty impressive field. I say pretty impressive. We've seen some truly mind-boggling fields of Riftia on this expedition, and this one is quite extensive and large with all kinds of size classes of animals. There is a small mirror flange in the middle of the screen. It's hard to see. It's quite small, and we can't really get under it for you, but we can try to show you. There's a little bit of discharge there coming out from underneath that rock, but when we get onto the other side of it, you'll see a much more dynamic feature. Starboard. We're going to swing around to starboard, and you'll start seeing, if you keep your eyes peeled, there's a mirror flange right in front of you. And what we're going to do is we're going to fly low and then high and give you a view of what the discharge looks like, and you can get a sense of how these features behave in the environment. There's some more spectacular ones on the other side. This is our first view of the mirror flange on today's dive. So these flanges accrete outward, and if you look up at the top left of the screen, you can see a small flange up there. So let's go up and down and give them a feel for what these things look like. So now you see the reflection of light, and as we move up and down, that light will appear to just vanish, and you can see the rocks inside, the shimmering pyrohotite that's inside and the underside of that rock, and it looks invisible, and that's because of Snell's law of refraction. The fluid that's trapped under that flange and seawater the light travels differently in those two fluids. So there's an angle where you actually the fluid appears to disappear and you can see through it. It's kind of like seeing your reflection on a lake, but if you look at a different angle, you can see through the lake into the water. So same phenomenon there. That we've almost got disappearing interface, and there you lose the interface and can see through it. So let's go up a couple and down a couple times. We're going to do some sampling here in a minute where we move up into the flow of that flange, and we pull a water sample to characterize its chemistry, and we're probably going to take the temperature of a couple of these flanges just to see. We've been getting temperatures around 366 degrees on most of these flanges. I think that's our high. On average, it's been around 170, but here's a really dynamic one with 3 different layers. Go back up there. Oh, wait a minute, go back down. That looked like stalactites for a second. Wow. Look at that phenomenon. That's outrageous. Whoa. That's new.
What's that?
Look at it. It looks like stalactites.
Oh yeah.
You know what I mean? It's not stalactites. Obviously, this is not a cave, but
under the water.
It's under.
Yeah.
It could be. It's the minerals. I mean, it's under the fluid. Yeah. But that's different. Every time we do this, we see something different. This is not an active chimney. This is microbial. This is an active chimney. Here, this is an active flange. And it's just because we're seeing the minerals that are under that water. And that is unreal. Wow.
That is pretty awesome. Do you want highlights?
Yeah, I think so. Because that's, I'm sitting here kind of speechless right now. I mean, those are minerals. Look at that. Those are rhombohedrals.
This is
of
exactly
crystals. That's unreal.
made a lot of amazing views.
I'm going to zoom in and try to get a better shot of those.
Wow. It looks like feathers.
Oh my goodness.
Wow. Highlights.
Jeez. Just when you think you can't be odd anymore, you see that.
Wow, look at the ripples now with it.
You could hear a feather hit the floor in this room. I think everyone is kind of speechless. This is pretty magnificent. I don't know what those are. I know they're minerals. I don't know what mineral. If there are any geologists watching the feed who have an idea what this could be, please do share your thoughts. We can have a contest. But my word, we've looked under a lot of these flanges and we've not seen anything quite like this before. My colleague Carlos from UNAM is saying that the minerals are dripping, and I absolutely agree. They're propagating downward, but these feathery patterns, I don't know what that would. And it's quite the contrast between the plumes of the riftia and the minerals, right?
That's so. Will the minerals actually come out of the water?
Yeah, but look at this. Look at the shape. Look at the texture, right? Geology and biology
It's amazing.
superimposed.
Can you explain what the riftia are?
So the riftia, the red in the bottom of the screen are the plumes of the giant tubeworm riftia, and riftia's plumes are they circulate their blood through their plumes. Their blood contains hemoglobin, and it's a hemoglobin that's very different from our own hemoglobin. It's very, very efficient at trapping oxygen, and it also binds CO2 and sulfide and feeds their symbiotic hosts who live in their trophosome. And so the riftia is sitting here, pulling out its nutrients from the water as material, flows from that chimney and is discharged into the overlying seawater. And you can see again, this is a very dynamic chimney or flange. There's 3 layers of flow here. This bottom layer that has just these spectacular mineral precipitates that look like feathers. And you can see that they are behind, a layer of shimmering hydrothermal water, the fluid discharge. And if you look on the lower right-hand part of your screen, you'll see a chimney that is no longer discharging at all, and that has a white microbial mat underneath it. You might think that the same is the case for the chimney in the middle part of the screen, but that one is actually still flowing, as you'll see in a moment when we start to rise up a little bit or come down.
Come down to get a lower one.
What rate do you think this is growing at, Mandy? Like, is that quantifiable? Have you seen a huge change since November?
This one has changed a good bit. I mean, it wasn't, we didn't notice the mirror. I don't know if the mirror flanges weren't here. I think we would have noticed them. I don't think they were here in November. I think this accreted. But this is that lower one that is not active, and that's clearly microbial and sulfur debris under there. The white gelatinous blobs that you see are precipitates of elemental sulfur that result from microbial sulfur processing around these features, and there's a lot of it that goes on here. Those invertebrates that you see, those scale worms, are around 3 centimeters, just for those of you wondering how big this is right now. But now we're in the part of the flange where it looks, and you can see here, there's these lots of scale worms on these rocks. They munch on microbial biomass. But as we go up and down in the flange, you'll get this sense of appearing and disappearing. It's like a mirage. It comes into view and then is obscured by the interface once we pass through that refractive boundary.
So I'm going to ask that.
Mandy, what's it like? I mean, you came here in November in a manned submersible.
Yeah.
I mean, these are pretty amazing images. Now, what was it like when you were sat in Alvin looking at these?
Well, seeing, recognizing, seeing this when it's 1.5 meters, 2 meters away from you is pretty spectacular. The only thing between you and the feature is a nice layer of titanium. But I don't know. I mean, the beauty of SuBastian is that we can just move up and down, and that's not something that's really that easy to do in the Alvin. You don't have the freedom to just sort of delicately dance up and down and check the features out. You basically, it's much more hard to go up and down because you've got to ballast with water. So we wouldn't necessarily have observed some of the things that we've observed here with SuBastian. This is just, phenomenal what we're doing right now.
I guess, as my old man would say, horses for courses. They both got their purposes.
Exactly. They both are, we came to Alvin Spire on this expedition because we had just sort of gotten a glimpse of it with the Alvin, and hence its name, Alvin Spire. But now we've learned so much more, and we have so many more questions about how these structures function. And there's, yep.
Give everyone a welcome.
Welcome to all the IFLS viewers. We're here in the Guaymas Basin in the Gulf of California, exploring these magnificent hydrothermal flanges and chimney structures that are all around us here. We've got 4 to 5 of these structures that we're going to visit over the next 4 hours or so. We're here right now at Alvin Spire. This is a chimney that's about 10.5, 12 meters tall and about 5 to 7 meters in diameter. It's a very large structure. This is the view you're seeing right now is hydrothermal fluid flowing, actively discharging from under one of these flanges. And if we move up and down a little bit, you'll see just an amazing example of Snell's law of refraction as the interface there appears and disappears as we move and migrate up through the water column, changing the incident angle that we're looking at the interface through. So while we're here, can we take a temperature in that lower and the upper?
So one of the first tasks we're going to do now is take a temperature reading.
Where the
using
feathery
a
minerals are,
temperature
yeah.
book. Some of the vents we've been looking at around here are 300.
And I want to get a water sample. I can't decide if I want to do it here or the other side. The other side doesn't have as many.
At the moment, Professor Joy is discussing with.
Dead? Oh, bummer. Okay. All right, we can't do that. So let's just fly up and down, and then we're going to position ourselves and collect water, but I think we're going to do that on the other side where that big flange is.
This is okay. There's a chimney to the foot in the rear as well. I'm sure what
Yeah.
that known is.
Yeah.
Can you set the big picture scene? Where are we?
Okay.
So
these
we're
are like-
here at Alvin Spire, and we're extending our examination of these amazing chimney structures. These large geologic features result from the reactions of hydrothermal fluid and seawater, which is precipitating out all of the minerals that you see in front of you. So this chimney is a feature that it only exists because of the interaction of these hydrothermal fluids and seawater. They consist of both iron sulfur minerals and carbonate minerals, as well as amorphous silica, barite, and a number of other really interesting minerals. You'll notice a lot of colors. We've seen beautiful purples and greens that are also minerals that we have not yet identified that we'll be identifying when we get back home to Athens, Georgia. But these features are truly magnificent. This is mineral precipitates under the flange where the hot fluids are reacting with seawater, and things precipitate out of solution because of the disequilibrium created by that reaction. Right at the edge of where the fluid flows in, you get these beautiful riftia tube worms. So they're just spectacular structures. So let's fly up and down and then head to starboard and do a roundabout. As we come around, you'll see there's a nice view of the silver looking interface, and you can see the discharge here. And these riftia, these animals are living, especially these guys living right under the shimmering ledge. I mean, they're living right on the edge of some super hot water. You see para-alvinelids in these rocks. These riftia are living right at the edge of where that, fluid, several hundred degrees, are just flowing right above their heads. Talk about hot-headed.
They can't straighten up, right?
They cannot. They are limited by the tube. Yeah. So the tube is hard. It's inflexible. That's the proper word. It's not. It's soft, but it's not flexible. Oh, is that what the ground fault was? Okay.
1, 2.
It's a bummer. Still with the CTD.
Yeah.
Live.
So let's, when we get over to the other side, on the back side of the chimney, we'll see that big spire feature, which is pretty spectacular.
Now we are monitoring the IFLS feed as well, so if any of you've got any questions, we'll do our best to answer them as well.
Can you talk a bit about the habitat that's being created by those animals?
So the animals themselves are a surface area for other animals to exist on. We have in this view, you can see the tubes of the riftia are made of chitin, and on those tubes, because they are a solid substrate, you'll see that microbial mats, these orange and pink beautiful structures inside here, they grow in amongst the riftia tubes, and all of these scale worms that you see are essentially grazing on the riftia tubes and consuming the biomass that's growing on those tubes. And the reason that microbes grow on the tubes is that the hydrothermal fluid is flowing all the time, but as a microorganism, if you're free living and you're just floating around in the water, you may or may not be in contact with hydrothermal fluid. The mixing regime is very dynamic. If you live on a solid surface like the tube of riftia or the tube of one of these chimney structures, can we back off so that we're not?
Someone was asking, how come, and it's a good question, if we're saying it's hundreds of degrees, the temperature on that screen is only showing 3.
The 3-degree temperature is the temperature on the CTD on the top of the ROV. So in front of us, under those flanges can be hundreds of degrees, but the background temperature is essentially, it's about actually 2.9. So 3 is a tiny bit elevated above ambient. But these animals can't exist in super hot water. So the most eukaryotes are limited to 40 to 50 degrees centigrade. So some of the para-alvinelids and alvinelids can handle high temperature exposure to short periods of time, but not for long periods of time. If you look in the screen now, you'll see a beautiful octopus in the down-looking camera. And this octopus, oh, I have to go down? Okay. You're telling me what to do and I'm not listening. There we go. There's your camera. Yeah. Yeah. So here's an octopus saying hello. There are a lot of octopus that live here. They're very shy. Oftentimes what they do is when they see the SuBastian, they flatten out, puff up, and they make their skin lumpy. So they'll change the texture and color. See how he's darkening? And he'll flatten out like a pancake and try to mold into the sediments, and his skin will become very lumpy. He'll get these indentations. There he goes. There's the indentations, and now he's flattening out like a pancake. He's trying to disappear into the sediment so that he becomes invisible to us. But of course, he's not going to become invisible because he's too beautiful. I don't know why they swirled their arms up that way, but they do that every, yeah.
I think they're
Yeah.
just getting into a defensive stance.
I think they're getting into a defensive, he's gonna whack us with his tentacles. But it's really beautiful. This guy's gorgeous. I don't think this is this is not the last octopus that we'll
Someone's
see.
asking how rare it is for humans to see this type of structure, like for people to visit this and get the chance to see this.
Well, extraordinarily rare. We didn't really know these were here until a few weeks ago, and had no appreciation for them. And for us, you know, for our research, we might get to come and sample these areas once every 3 to 4 years. We get that opportunity, so it's you know, we treasure the opportunity and we want to take full advantage of it when we have it.
Okay, we're just going to move the vehicle slightly, so just bear with us a second. We're going to try and get some of those nice images. At times the audio may drop in or drop out as we discuss our plans in the control room. Yeah. At the moment we've just moved slightly off the spire. Just hang with us a moment, and we're going to move to the southwest side. Today's dive, the mission objectives are to take fluid samples from these areas as well as other biological samples. And also it'll be cool having you guys along for the ride. We'll be taking some nice imagery. And if you've got any questions, you can ask on both our Facebook page, the YouTube Facebook page, and we're also picking up the IFLS feed as well. So we'll do our best to try and answer your questions. Today you're joining us from 2,000 meters below the Gulf of California. These images are coming to you from remotely operated vehicle SuBastian, owned by Schmidt Ocean Institute. Nobody's actually in this vehicle. We're all sat in the control room on research vessel Falco, 2 kilometers above SuBastian. The commands our pilots are sending are going through 7 kilometers of steel armored umbilical. So today's mission is, as I said, fluid samples, but also there's an exploration element. We're going to look around this vent field. It was previously mapped by the autonomous underwater vehicle Sentry, operated by Woods Hole, same people who found the Titanic, and we're using some of their maps and exploring some of the lumps and bumps and seeing what's here. So stay tuned. At the moment, my colleague is putting a map up where in the world we actually are. So while we're just transiting around the other side of the spire, you can see here's the Gulf of California, and we're zooming in on the ship's position. We're currently at a place called Guaymas, and this is known as Poseidon's Pagoda vent field. And as we get closer in, you'll see maps. Now these have been generated by the autonomous underwater vehicle Sentry.
Oh, trash.
Actually,
trash.
those on your screen, while we're slowly going around the vent, you'll see that there's plastics here. There's a lot of junk around these vents. Now bear in mind, someone was asking how rare is it for humans to see this? Very rare. But what we've learned is that there is a lot of junk around these vents. So humans' impact is already getting to this location, even though this place hasn't really been visited that much. And while we've been doing our work here, we've seen lots of plastics and lots of balloons, just rope, just general detritus of humankind, littering with these amazing and unique ecosystems.
Let's swing a little bit more to starboard before we start heading up.
So here we go, we're going to carry on around this vent. At the moment we're at the bottom of the vent, but you can still see there's still a lot of bacterial mat and flock in the air. And there's still some more plastic bags. There's a black bag just on the feed, just down the bottom of the screen at the moment. And I'd like to say this is only one of a few we've seen, but we've seen hundreds, if not thousands, during the course of this expedition. And let's see if we have any questions on our feeds.
Oh, there's more. There's an octopus.
Oh, yeah.
There's another octopus. They
Oh,
hide
there-
in these little nooks and crannies in the rocks. There's one that is.
So yeah, it's coming up on the feed. A lot of people are talking about the plastics we're seeing at the moment. So if there's anything you guys take away from this, apart from the absolutely cool ecosystems, take away the fact that even here, there is a huge impact that humans are
Let's
making
continue.
on these amazing ecosystems. The rubbish that we throw out when we don't recycle, and all the stuff that goes to landfill, a lot of it can end up in the oceans, and you see it here. So please bear this in mind, the impact we're having on the planet. Tell your friends, share these images with them. It's disappointing to see it.
How big is the vent?
How big is the vent? Actually, I think you better ask Professor Joy that. I don't,
So these features are occurring along fault networks, which sort of dictate where the discharge occurs. Any one of these structures, I mean, Big Pagoda is 23 meters tall and 10 meters in diameter. Ace's Spire, the Alvin Spire is on the order of 10 to 12 meters tall and probably another 10 to 15 in diameter. These are large structures. We're going to sort of continue to the starboard and up the face of this thing. So we're at the base of the spire right now, and we're at a depth of 20 meters. So let's just go up to the top and see what we get. I think we're going to probably end up at about 2,000 meters when we're at the top. Maybe only 2005, but it's a pretty tall structure. The bottom of the structure is glass sponges, and can we continue around? Yeah.
someone's asking why we
right
don't
here.
pick up the rubbish. One of the main reasons we don't pick it up is it also poses a threat to the ROV. Not only does it pose a threat to the animals that are here, if we get one of these plastic bags in our
Yeah,
thrusters,
so right
it's
in
mission over. One of the more important
So it's-
things that we can do here is educate. So the images that we collect and we stream out to you is important to get the message out. We could spend hours collecting this rubbish here and only get a fraction of what we've seen. Instead, we're going around documenting these ecosystems, but getting these images out. This doesn't take an ROV, which can only pick up several pieces of rubbish per dive. This takes a bigger effort. This takes educating people on the effects of rubbish in the ocean.
So now we're heading towards the front side of the mound where the most active discharge is. And you'll see this is a pretty actively discharging area right at the base of the structure on the front side. There's microbial mats, tube worms. The white and orange patches are microbial mats. The Riftia is very happy here. It looks very healthy. And as we go around the structure, we're gonna start to go up. You'll see little squat lobsters and scale worms on the surface as well. They basically wander around the surface and consume microbial biomass. Now we're gonna go vertically up the face of this to get to the top of the spire, and you'll just notice that as we go up there's a lot of activity on this structure, a lot of animal biomass. These are not the happiest looking Rifty I've ever seen, but
Someone's asking what the typical type of trash, the most common that we're seeing. I'd say plastic bags.
plastic bags and balloons, believe it or not.
Yeah.
We've seen a lot of balloons.
We've also seen, I think, like bits of rope, which might be from fishing, but a lot of plastic bags, and they drift with the currents. As we know, in shallower waters, they pose a hazard to things like turtles, which mistake them for jellyfish. But down here, they just foul up on the ecosystems. We've seen the remains of squid caught up in what we think is rope. We also see lots of nets.
Continue
We've seen
going.
nets on the
Yeah, I
seabed,
think
and
we
Falco
want to
has
be sort of on
recovered
that side,
several nets
straight up.
previously that we found floating in
Yes.
the ocean. And these are brought on board, and then we dispose of them. But there, nets also pose a major risk, along with long lines to the ROV. Getting entangled to them is not a good day at the office. As you can see, this is a truly spectacular, amazing ecosystem. But just 4 meters below us is a trash heap.
Oh, there it is. Okay. This is the top of Alvin Spire. Here we are. Pretty impressive. This is a tower of freshly precipitated chimney material. This was not here before when we were at this site. If we go up and down here, as we're going to do over the next couple of minutes, if you look under those ledges, you'll see the beautiful mirror reflection that we spoke about before. You can see the color here, and as we continue straight down, that will disappear, and you'll just see up into that cavity and get a sense of what kinds of minerals exist in those rocks. They'll flash and glimmer at you. And directly in front, on the face of that rock, reappearing structure. This is one of my favorite sites that we've been studying on this expedition, because you've got that giant that very large reflecting pool there at the top of the chimney. It's just truly spectacular to see this in real time.
So someone's asking, is this actually like a type of volcano, and where does the water that we're seeing come from?
Oh, that's a great question. This is an underwater volcano, and this water comes from deep circulation of seawater through the crust, and that water is superheated as it travels through the crust. It's chemically altered, and then it discharges because it's warm, fresher, and buoyant. It discharges from weak points in the crust along fault networks at features like you see here, and all of the mineral precipitates that you see in front of you are a result of these rock-water interactions that are happening, and chemical interactions that happen between the seawater and the venting fluid as the fluid is expelled from the seabed. So these chimney structures can accrete very rapidly. They can grow in some cases, many inches, if not meters a month. They're fragile. As you can see, the geometry is a little bit mind-bending. How are they stable? You have this little pole in the middle with this flange coming out to the side. How does that stay stable and not fall over? Well, it does fall over at times. We find a lot of chimneys that have sort of tumbled over to the edge, and you can see a new flange is forming right up at the top of this structure that's beginning to grow laterally as well as vertically.
And the species we see here, why are they so unique, and can they exist off the vent that we're looking at?
No, these animals derive their basic nutrition from the chemicals that are in these vents, and they are chemosynthetic, chemoautotrophic organisms. So they use chemical energy to generate their biomass, much like photosynthetic organisms use light energy to generate their biomass. Chemosynthetic organisms use chemical energy to generate theirs. So if you move away from an area of hydrothermal input, you don't get these animals. You only get these animals in association with these vents. That's why these vents are called oases in the deep sea.
Crazy that it's staying up. It looks like it's on a hinge or something.
It does. I mean, this one is mind-boggling to me because it's coming off a pinnacle and then off laterally. It's like, how is that possibly stable? Can we do go down a little bit more so people can get the sense of what's underneath there?
So we're just going to bring the vehicle a little bit deeper so you guys can see these
there we go.
mirror flanges.
So
Yeah,
you said that the animals could not live in that really hot, shimmery water under the ledge, but there's a lot of stuff that is alive in there, right?
Yeah, and those are all hyperthermophilic microorganisms, and they like it super hot. These hyperthermophiles can live at temperatures greater than 70 degrees centigrade, and they thrive at high temperature. They have special enzymes, special lipids in their membranes that keep their cells intact, and they are very acutely adapted to surviving in these really challenging habitats.
So is there anything we can learn from these vents or take away, like bacteria that might be helpful in like pharmaceuticals, or is there anything useful here for us?
Absolutely. I mean, one of the reasons that we're really interested in these organisms, aside from just a purely sort of evolutionary biology standpoint, is because of the natural products that they utilize to succeed and thrive in these habitats. So, it's membrane proteins, metabolic proteins, all of the metabolic machinery that they utilize to impose their will on the biogeochemistry of these systems is really useful for us to study to potentially, there might be new antibiotics here. There might be cures for diseases hidden in these organisms, antivirals. There's just a number of aspects of natural products chemistry that are quite fascinating to pursue in these types of habitats. So here's a spectacular view of just the microbial biofilm that's growing on one of these relict flange structures, and you can just see lots of different colors, lots of different textures. I'm going to zoom in so you can see what these mats look like. They are lush, filled with life. There's some folliculid ciliates in here, the little white-tipped organisms, but a lot of this biomass is microbial. This is Beggiatoa, which is quite just incredibly abundant in these habitats. And over to the other side, you've got different colors of Beggiatoa. There's orange and white and pink. And then there are these really weird iridescent purpley blue biofilms that we don't know what they are. But yeah, I mean, you could spend, probably years studying these systems. Say again?
Up and to the left, we can look up into that
Oh,
second.
So just moving the camera. One of the questions that came in is, people are considering mining these. Is that a bad idea? And what sort of minerals are commonly found in a deep sea vent like this?
So there are a lot of valuable rare earths and trace metals in these in these chimneys, and they're quite concentrated in these chimneys because it's primordial fluid. It's very rich in valuable metals. However, as you can see, these are very localized features that support a dynamic and thriving ecosystem. So I personally am not a big fan of the idea of mining in the deep sea, because I think that we humans might be a little bit better off if we thought about more efficient and effective usage of the materials that we have, as opposed to just constantly looking for new resources, making, recycling and reusing some of the resources that we already have. These structures are, they're very localized. They're very hard to get to. They're at the bottom of the ocean, and it's unclear the damage to the system that would be done if you remove structures like this. It would be immense. Here's a close-up again of one of these biofilms, and you can if you just look around, you could describe 20 or 30 different types of organisms, including a Para-alvinella. That's the little tentacle-y thing you see at the bottom here. In this view field, and we don't know what a lot of these things are. We haven't even identified them yet. So I think we really need to understand and have a full appreciation for these systems before we consider mining them to get trace metals and rare earths.
On that question as well, someone was asking, and I know you're busy, Professor Joy, there's some talk about these being present in the rest of our own solar system. That NASA's got various missions to other planets where there's been seen to be like jets of water shooting out from other moons.
Titan.
Do you think that this could be a similar ecosystem or similar feature could be found on another planet within our own solar system? And does studying these help our understanding of things that could occur on other?
Absolutely, that's a great question. So when you think about life exists because of life takes advantage of disequilibria. So here we've got just an astonishing example of disequilibria, hot water mixing with cold seawater, which offers these organisms the opportunity to live at this interface that's rich with both food and oxidants. So we breathe oxygen and we consume, pizza or spaghetti. And these organisms, they breathe oxygen per se. Some of them can breathe nitrate, but they use for their energy source the raw materials, the raw chemical materials in this fluid: methane, ammonium, hydrogen sulfide, molecular hydrogen, carbon monoxide, reduced iron. They take those raw chemicals and turn that material into metabolic energy. The same sorts of processes could happen in other worlds where there's a source of energy and a source, they need oxidant and they need reductant. They need a source of energy, which is the reductant. They need a source of electron acceptor, which is the oxidant. And these sorts of habitats are just a prime search ground for new metabolic pathways and mechanisms that we might look for and find on other planets, especially those involving, metals and methane and things of that nature. That's one of the things that we're really interested here in Guaymas Basin, is understanding how methane is cycled, both produced and consumed by the microbial populations, because that's a really good metabolic target for looking at other worlds like Enceladus, Titan, or even Europa's ocean.
Cool. What should we look at next?
So I think we should, let's fly up and take some CTD temperatures on the vehicle in the exit. Where did my magic point
Okay,
go?
and John's gone back to showing the overview of our
we
location.
could go sit up here, and maybe we could show them the down-looking camera while we're sitting up on top of it.
John,
I want to try to get in the effluent stream and the turbidity stream of the structure. There's a nice view of the fault that runs up the base of the structure. You can just see it cracks up on the edge, and we'll show you the big fault in a second when we fly off to our second target. But these structures are they're just magnificent to see, and it gives you an appreciation for how microorganisms can take advantage of the most extreme conditions to just thrive and proliferate and generate phenomenal amounts of biomass. We're gonna trip 2 Niskins in that turbidity flow. I wanna make sure we see if we could get a temperature assessment and then turn and trip the Niskins in the same sort of feature, that would be good. I think we did it in this little guy before.
Professor Joy's
but I'm
saying
not
about
sure.
tripping Niskins. On the side of the vehicle are 4 bottles. We can close these using the arms, and these will trap water from the surrounding area inside the bottles. These will then be brought up to the vessel, where they'll be studied and filtered by our scientists to figure out what's living in them, what minerals are in there, and they'll give us an important insight into some of the fluids coming out of this vent. I like the little vent to the left. Looks like it's got ears and a nose.
Yeah, it does, and it's a little like bunny.
Yeah.
There's a bunny. This is like looking at clouds. You can just imagine all these different structures. There's a bunny. There's pig. What do you see?
What do I see? I don't know. I need to see a doctor if I say what I see. But of all the vents you've seen, what do you think of these ones? Do you think they're some of the most spectacular, or are they just run-of-the-mill vents?
No, they're spectacular. I mean, the geometry, the delicacy of these structures, the fact that it's not just a rock, it's a living thing with life. You look at any substrate on here, if you zoom into anywhere, you're not just going to see bare rock anywhere. It's all covered with biology, and I think that's what makes this particular area really unique. I mean, all at all hydrothermal vents, you've got dynamic microbial populations associated with the structures, but this is just vivid. It's in obvious detail.
So hydrothermal vents
Whoa,
found
3.8.
elsewhere?
Whoa.
Ooh.
So the temperature we're measuring the temperature that you see on the screen now is the temperature of the vehicle CTD in the flow, the effluent stream from the vent. So we're about a degree above ambient now. So that's a good chunk of vent fluid, about 4.1. Ooh, this is even better. I think we can hit 4. I think what did we get last time? 4.4.
We've got up to 7 on the vehicle before.
Yeah, that was a big pagoda though. I think.
Yeah,
I think
yeah.
here we got about 4.2 or 4.3.
I just saw 4.3. Yeah.
Oh, 5.
So these temperatures probably won't sound much to you, especially those who are currently freezing in the United States. But actually in the deep ocean, this is quite warm. But also if we took our temperature probe and put that into the vent, we'd be seeing something like 360 degrees Celsius. This dissipates really quickly, but still having 5 degrees Celsius in the deep ocean, it's quite a big thing.
So let's think about.
So if you orient the port side over.
Okay, someone asked how much is 360 degrees in Fahrenheit? That's
Multiply by 1.8.
512ish. Yeah.
that's 660, I
Yeah.
think.
How much was it again? 300?
360 degrees.
Alright, so 720 minus
Doing some maths.
72.
There's an app for this,
720
Chris.
minus
68, 680. 680 degrees Fahrenheit.
There's
It's
that,
hot.
right here.
That's the human calculator. We don't call them Google for nothing, you know.
Yeah.
So in the control room at the moment we have 4 people, well 5 actually, 6. We've got Professor Joy who's narrating the dive. We've got our pilot Russell who's piloting Sebastian, our co-pilot Chris who's going to get the arms out momentarily and sample one of these water bottles. And then at the far end we've got John who's working the live feed, monitoring your questions and making sure that the data coming from the vehicle is logged. In the back we've got our data logger Carmen who's making sure that the dive is all logged. At the moment you'll see the basket coming out on the vehicle. We have basket, tool tray. It's got multiple names. On this we keep the equipment that we need to complete this dive. At the moment we're just putting it out so it's out of the way of the arm as we try and trigger one of these water bottles which will take a sample. Now these are connected to the 4 rope bowls that you see here. When we pull one of these with the manipulator it causes the spring-loaded bottle on the side of the vehicle to snap closed, trapping the surrounding water inside it. This is then brought up to the ship and studied by Professor Joy's team. Now, the arm that you see coming into your screen, this can lift up to about 200 pounds, I believe. And on the ship, our co-pilot is controlling this using an exact replica of the arm, what is known as a master controller. And as he moves each section of this arm, the corresponding section on the slave on the vehicle Sebastian, 2,000 meters below us, moves. The arm also has buttons to open the jaws, which you see on the screen now, and rotate the wrist. So our co-pilot is currently controlling this on a vehicle 2 kilometers below us, but the commands he's sending to it are going through our umbilical, which has several kilometers of cable. Now, remotely operated vehicle Sebastian can dive to 4,500 meters, but we have a little bit extra cable. As you can see at the moment, they're rotating the wrist, and we're going to grab the white rope bundle there and pull it. This will cause one of the bottles to snap closed.
For sample.
At the moment, our scientists are making a note.
Yep.
It's important we know exactly where these samples were taken. I think we're going to pilot the vehicle back towards the plume. So while we're waiting for the first sample of the day, these images are coming to you from remotely operated vehicle Sebastian, operated by Schmidt Ocean Institute. The vehicle is being operated off research vessel Falco in the Gulf of California. The vehicle is 2,000 meters below the ship, and it's remotely operated, so nobody's sat
Are we in the
vehicle.
turbidity? Yeah.
Everyone is sat
Perfect.
comfortably
Yep, I
in
can
the
see it.
control room on the ship.
We're gonna pull 2, but just 2 because we need the other 2. And let's see which one it is.
We're just letting Professor Joy oversee the first sample of the day, and then I'll get her to explain exactly why we take these water samples and what happens to them when they get on the ship.
All right.
Next one.
Which one was that?
So that was the first sample taken,
And move to the next one.
and now we're going to move to the next water.
It misfired?
Yeah.
Jeez.
We're getting information that unfortunately bottle may have misfired. We'll try the second one. We'll get back to some of these images shortly.
This gives you a feel for the technical sophistication required to sample these systems. We can guide our sampling very carefully from 2,000 meters
this
above
dead?
bottom using the cameras and sensors on the vehicle. And we can position ourselves just so to get a sample of this plume water, which is what we're targeting. Oh, we're right in it now. You can see it, yeah. We're really interested in the microbiology of these plumes, and we want to get a feel for and a quantitative assessment. All right,
Number
This-
4. Number 4.
number 4. Okay, I think we're only going to be able to get one here since we lost one. Are we good on that number 4?
Yeah, 4 is
Okay.
right. You clear on that?
So when we get back, when we get this water back up, we'll measure chemical signature of the water. We'll filter some water for DNA, and then we'll use this water for inoculation and enrichment of novel microorganisms. We're quite interested in the organisms that process methane and sulfur and ammonium, and we're looking for organisms that consume these substrates, and these plumes are a great place to find novel organisms that cycle methane, ammonium, and ammonia and hydrogen. All right, what else do we need to do here? We need to, I think we just need to fly up and down a couple more times, give people a good feel for, get some good photo doc on this feature. You can see, if you just look at the base of the structure, several venting orifices here at the base of the chimney. Then these series of mirror flanges. We're going to go up and down one time, then we're going to drive down the big rift in the back of the structure, which is back here. We'll go this way. And then we're going to head down the fault line towards what we call the trench of trash, which I think is a really good thing for people to see, because it sort of will give you an appreciation for the impacts of human sort of wasteful treatment of trash or negligent treatment of trash. That this stuff can just end up concentrated in a hydrothermal field in the Pacific, in the Gulf of California, off the Pacific Ocean, is just it's kind of mind warping. Here we've got a view of a whole number of these amazing mirror flanges, left to right. Your screen's full of them. And as we go up and down ever so slowly, you can see the interface just disappear and reappear between your eyes. It's amazing. You know, when I go to sleep at night, I see the water flowing. Do you see that water flowing in your eyes?
It's trippy.
It's just it's
makes it
seared into my memory.
It makes things look like a kind of abstract painting when you
Yeah,
close up.
Jackson
This
Pollock.
one here
Yeah,
is always-
yeah. This one is amazing, and this one too, because of all these layers. It's just this three-dimensionality of it is.
So if someone said, "Oh, there's nothing at the bottom of the ocean," it's there's nothing down there. What would you say to that? Like, what?
Ha 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 ha! Don't be silly.
Someone asked, do they play an important part in the ocean? Is it an integral part, or do we not fully understand what these vents are doing for the oceans? Are they turning something over? Are they doing something for the oceans?
They do a lot of things for the oceans, but a real obvious example is the global magnesium cycle. Magnesium, might not seem like an interesting thing to you, but if the magnesium is a critical component of our own human physiology. If we don't have enough magnesium, we get cramps in our muscles, we don't sleep well, our brains don't work well. Magnesium is critical, and chemical oceanographers were not able to balance the global magnesium budget until we found hydrothermal vents and discovered that these vent features are a very significant sink for magnesium. So they play important roles in global biogeochemical cycles of things like magnesium, of trace metals. They're inordinately important, and not just because of their relevance for astrobiology. These systems are a very critical part of the global ocean ecosystem, and they play a dynamic role in global biochemical cycles. They also allow us to evaluate and understand processes that are really hard to measure in everyday average places because they occur at a slow pace. They occur at an accelerated pace at places like this. So it's a way to really look for processes that are new and unique because they occur at rates that you might not be able to even detect them in other systems. Wow, this is just
People keep asking the question, why is it just the hemoglobin or the blood inside the tubulums? What is the importance of color at this depth?
all enzymes, the hemoglobin and other critical enzymes, cytochromes, for example, have metal enzyme active centers, and those metal enzyme active centers are often colored or engender some sort of color. to the enzyme. And every color that you see on your screen, some of it is mineral, but the orange microbial mats, the red riftia, some of the purple bluey microbial mats, the pink microbial mats, those are all pigments that these organisms have, enzymes that they use to carry out their metabolism. And so if you think about it that way, life is color. Color tells you a lot about where to look for life. So if you see something super colorful and you realize it's not a mineral, then it's probably an interesting pigment that you could take a sample of and evaluate. All right, this is beautiful, but we need to move to our next target. So let's come up and move to port.
If someone was asking about the date on there, we'll just confirm. Someone said 2019 03/10.
That's the 10th of March.
It's
yes.
the 10th of March.
Yes.
At 1610, because we operate on UTC.
Yeah.
So anywhere we're in the world, we reference to UTC. If we switch between local times and we move from one zone to another, it would cause us all manner
We
of
would
problem.
all get very confused.
So we log to UTC, even though we're in the Gulf of California and the time zone may be different.
And when we're on ships, we live in Zulu time so that we can be consistent. So here's a patch of really, can we hold steady for 1 second? So here's a really great example of what I was talking about color. If you look at the screen right now, you will see the pink and the purple and the white and the orange. Those are all distinct types of microbial biofilms that are growing on these rocks. And we know what the pink and the orange is. We don't know what that white patch is. That is not Veggiatoa. We think that's an epsilon proteobacteria. And we don't know what the bluish biofilm is. And the little red tintly things that you see, those are para-alvinellids. Those are also two worms similar, of the variety sort of riftia, but they can handle much higher temperatures. And then if you look even more carefully, you see there's gastropod snails living on the surface. There's the gastropod snails, and then the scale worms in the background. So there's just a whole variety of life, all different colors, that are living on these structures.
I've got a good question for you. These were only first identified, say, 30 years ago. How did these go unmissed for so long, considering they're relatively new to science?
So I don't know. I don't know. I don't know. I think maybe they only popped up here recently. Maybe they were small structures and the big ones just, popped up recently.
But the whole idea of like hydrothermal vents in the first find is in our own lifetime.
Oh, absolutely, and that's astonishing. And these vents were discovered because of heat flow measurements in the deep sea and the observation of heat anomalies. And it's like, where's that extra heat coming from? This is really strange. And then they take the Alvin down there, and holy cow, they find these vents. So this is the big fault. Can we sort of face this and back this way? What can you back? Yeah, your positioning. Yeah. So we're going to move down this fault now, and you'll see in the throat of this fault, there's a lot of diffuse flow that occurs along the walls of the fault, and there's rifty in there. You'll see colorful mats, and you'll be able to see the fluid discharging along the walls as well.
So at the moment as well, for those watching the feed, we're just showing the location and some of the multi-beam acquired data from the vessel in the picture-in-picture. So what you're seeing here is a 3D image of the seabed below Falco, and the little yellow vehicle is SuBastian. So this is us tracking the vehicle. For this data, the background map was acquired with an autonomous underwater vehicle, which maps the seabed to a few meters of accuracy. And one of the important things my colleague brought up, another reason why these vents weren't known, is we know so little about the world under the ocean surface. Only something like 10% of the world's oceans have been mapped to a high resolution. So there's still a lot we don't know about the oceans. And missions like this, the one you're currently observing, these are going out and studying what's down here. Exploration at its finest. Now, Schmidt Ocean Institute, one of the things that they do fund is more risky science, coming out to locations where we traditionally wouldn't conduct science. So this is a major positive. A lot of governments in recent years have been focusing on science with known returns, especially due to austerity. So the fact that we go out fund higher risk science as well, using various equipment, this is a major plus.
It is, because the opportunity to go out and explore places and find new things, that develops, research opportunities and portfolios that can extend for a decade. I mean, I think I'm quite certain we'll spend the next 10 years studying these systems and potentially develop blueprints for finding similar systems in other places once we get a better understanding of them. So it's really exciting to be a part of such an exploration and discovery based mission. And we are so grateful to the support of the Schmidt Ocean Institute and the staff and crew of the Falco and the team that leads and guides all the shipboard science and the ROV group here. It's just been a real pleasure to work with everybody over the past couple of weeks as we have made just stunning after stunning after stunning discovery working in these systems. So now we're moving away from the active part of the fault down to the inactive part of the fault, and we're going to transit now to a second feature. So we're
Trench of trash.
yeah, we're gonna cross over. We're gonna follow this fault line down to Falco's Fountain, and in a couple of minutes, here's an octopus. We'll walk you through what we're seeing and watching. But we're gonna, these features that we're seeing, are a few hundred meters apart. So it's a bit of a commute for the SuBastian to go from one to the other. But when we were making this commute the other day, we happened across a trench along this fault line, and what we've noticed in our time here is that a great deal of trash accumulates in these topographic lows on the seabed, and that's just because of how the currents are flowing and moving stuff around. But I, for one, was simply I was stunned by the amount of material that we're seeing in these trenches and along the seabed. And these aren't trenches in the sense of the Marianas Trench. This is a trench a few meters to tens of meters deep, but it's accumulating an enormous amount of debris.
And we know from recent studies and
let's
some of the work
head
done in
this
the
way.
deep
Down
parts by
trench of trash.
Alan Jamieson at the University in Aberdeen, that plastics are getting even to the deepest parts of our planets, down to 10,900 in the Mariana Trench. So it is really playing a significant role. And coming to a place like this and actually seeing the amount of plastics on this ecosystem was somewhat depressing. You've all seen just now the amazing images that we're seeing. And in a moment we might see some not so amazing imagery as we transit to Falco's Fountain, which we found and named the other day. So yeah, we're finding features like Falco's Fountain, but amongst all that, we're finding rubbish.
Lots of rubbish. And as we said before, one of the most common things that we see is balloons and various other plastics. It's just amazing how much stuff there is along the bottom that's trapped along these features. So we'll probably have some rather dead space as we travel along till we get on top of this trench. You can watch the fault line and track it just as we did. One of the ways that we found some of these new features is we just marched up the fault lines, and there's just feature after feature after feature occurring along these fault lines.
I guess while we're transiting to the event, John, if you want to show people where we are in the world, we can just explain while we're going across some of this a little less featureless seabed. So my colleague John has just placed on our screen an overview of the Gulf of California. And as we're talking, we'll zoom in on the work area. This area is known as Poseidon's Pagodas, the actual vent field. As you can see there, we're zooming in. And in a minute, you'll see some blue data. This is actually multi-beam data. So data acquired using sound from under the ocean, and allows us to three-dimensionally paint an image of the seabed.
There's the trench.
Oh,
Yeah.
there's a big crab as well on the image there. And here you'll see the white line coming down from the surface is showing where our vehicle currently is, where SuBastian is on the seabed. And the white track is where we've been wandering around this dive. So you can see there's features there on the seabed. And as we zoom in, SuBastian is approaching this trench area, this fault line that we're going to explore momentarily. We track our vehicle as well as three-dimensionally during the dive, so we can see where it is around some of these features. Unfortunately, there's no one to ask directions, so we need this tracking system. Stop and ask the nearest octopus. So in a moment, we're just bringing the vehicle closer down towards the seabed.
We're almost to the trench of trash.
There's another octopus. There seem to be a lot around this
There are
area.
a lot of octopus
Do,
here.
do they venture into the vent field to predate on anything?
They do. They'll get on the edge of the. I mean, there's actually, when we were at Alvin Spire in November, an octopus followed us from the bottom to the top, and I didn't see it until I was looking at the pictures later because it was behind that shimmering water cloud, so it was obscured. And it was really crazy, and I thought I was, hallucinating. That wasn't really an octopus. And then I had him in about 50 images that I had shot from inside the vehicle. So it was definitely there, definitely keeping us company, keeping an eye on us, making sure we didn't do anything they didn't want us to do.
So before we get to the trench of trash, can you tell anyone what to expect when we arrive at Falco's fountain after we've gone along this fault?
Falco's fountain is a spectacular feature. I mean, and this is another pagoda tower that we discovered by just following this fault line, south of Alvin Spire, and it's a truly magnificent flange. I think it's probably second only to Big Pagoda in my mind. It's just an amazing feature, and each one of these is unique, as I said before. But it's a big structure that we had a contest online to name it, and we're actually going to go to a new field next, so we'll have another contest online to come up with a name for that. So be thinking of what is that? That is
that a first bit of plastic or foil or something?
it's hard to say.
flock.
I think that's
Flock.
sulfur flock. Oh no, that might actually be, yeah, that's sulfur flock. That's elemental sulfur that's just wafting around. There's a lot of that. But when we get to this area, you'll be really surprised, because it's just, I mean, all of us were just in shock by the amount of just utter garbage that was in this area when we came upon it.
While we're going there, someone's asked, have you found what you're looking for on this cruise, Professor Joy?
I found more than I ever thought. I mean, I knew that we would have a productive expedition, and I knew that we would find some things that were just truly remarkable. But Tom asked me a few months ago, he's on board here doing a lot of the science communication. He said, what do you think you're going to find that's really cool and interesting? And I said, I think I gave a pretty waffly response, and I said, we're going to find something that is an amalgam of what we know about vents and vent structures, but it's going to be so different that our minds are blown. And I think that's a pretty good description of what we found. We weren't expecting to find these just tremendously complex features like the pagodas. And I think what I sort of had in my search image was a little mound that was maybe 2 meters tall. I was off by an order of magnitude or more. So we have found more than we ever dreamed of. But the saddest part of it is what we're about to see, which is this trash in the seabed. So we're now in the trash trench, in the garbage trench, or the trench of trash. Let's
get a human impact mark on there.
Yeah, this is some plastic. It's someone's bag of garbage that they actually dumped over the side. So that's actually an intact trash bag that's been on the seafloor for a while. You can see material growing on it. We do want to collect some of this coral. And what should be here are these coral tumbleweeds. That's what I call them. We think these are corals. They are inhabited by shrimp. We find them in these topographic depressions. This should be a lush habitat for these shrimps and other animals, squat lobster, etc. But what you see instead of just the natural system is all of these plastic bags and trash. It's truly just depressing and stunning. Yeah, we're going to sample here. But for those of you, wondering whether your trash that you accidentally, the bag you don't chase down and catch, what happens to it? I'm not quite sure how you accidentally drop a bag of trash off. You know, that's a purposeful discharge probably.
Yeah.
But a lot of this material, you know, just does not belong here, and it's a signature. It's a fingerprint of our activity and how we're just changing things even in the deepest parts of the
Do
ocean.
you find it really depressing? I mean, we've moved 50 meters off what is a amazing ecosystem. All those spires, all the riftia, we just saw those amazing tube worms, these truly spectacular sites which our viewers at home have just gone, wow, this is amazing, this is out of the world. And we're seeing the reaction on IFLS and our own feeds. People have just been blown away by what they've just seen, and now we've just brought them here. I mean, we went from amazing to trash in moments.
Yeah.
And what do you think of that?
And that's what we've seen again and again. We move. We either see something, we see garbage at the base of a phenomenal structure, or we're moving from one structure to the next, and we just see garbage after garbage after garbage. I would try and get.
So at the moment, the vehicle's landed on the
just,
seabed, and
you
we're
know,
just
a
about
good
to take
handful
some
of it,
samples
just break
using
it
the
off.
manipulator to study what this actually is. There's been some debate throughout this expedition what these actual tumbleweeds of either coral or have they actually come down from the surface like the trash. So we're going to take a little sample of this and we're going to study it. One of the benefits we talk about damage to the seabed is 20 to 30 years ago, a lot of science would have been done by trawling. And we know now that trawling is quite destructive to the seabed, or well, very destructive. So vehicles like SuBastian can go down, and the manipulator arm you're seeing at the moment can take little samples, just breaks little bits off, and leave the rest of the environment totally intact, as opposed to what trawling would do to it. So vehicles like SuBastian are really awesome tools for science. As well as the manipulators, as you're seeing, one of the best tools on this vehicle is the cameras. These 4K cameras that you'll see in the video image from, these can get the images to you and you can see them. We're using the ship's internet link, which I guess you think is amazing, but compared to your home internet, it's only 7 megabits up and 7 megabits down. And at the moment, it's all being used to get these images to you. As you can imagine, we're not towing a big cable around, and sometimes the signal may drop. So as Professor Joy was just saying, she's busy making notes on the sample. If one thing you take away from these dives is the amazing scenery that we've just seen now, take away the idea of all the trash that's on the seabed. Have a think about how you yourselves recycle. It's cool enough to come and see and explore the oceans, but they are turning into somewhat of a dumping ground at the moment. So if you get the chance to lobby someone and reduce that trash, or just change your own mindset, do it.
Just light level a little bit. As it's a larger sample, would we be okay using 3 and 4?
Yeah, that's fine.
I'm going to fill up.
So what's happening at the moment is the arm has collected a sample of this and they're now going to put it in a storage tray on the front of the vehicle. This is a retractable drawer. On that we have all our tools and science samples for the scientific samples for the dive can be put on there. As we lift off now, you're going to see a little bit of the trash that we've just been talking about. Those black bags, all those plastic bags in the distance. Ironically labeled plastic bag fish. So one of the things about this area as well is the amount of detritus in terms of biological that gets to the seabed. Every time the vehicle touches down and we lift off the seabed, it kicks up a big cloud of dust.
There's a couple signs for some of the recruits.
Pardon?
There's some signs for some of our
Oh, yep, we have some signs if anyone's on our basket. A lot of these are for the families of some of the scientists and crew on board. Now, our team here are often away for 2 to 3 months at a time. Our scientists up to a month, so they miss important family events at home, birthdays, and just generally miss their families. So a lot of the time, the basket of the vehicle and these sample boxes you see opening and closing at the moment, they're decorated by our scientists and crew to send messages home. And I believe that's our principal scientist, Professor Joy's family. Is that right?
Today is Jesse's twins. My twins and my older one were the other day. and I'm not sure, that's some meme that's very popular right now, but I'm not
We are going down
with
streets.
teenage meme. Yeah,
I have
what's
no
that?
idea.
I don't know what that means.
We're not hip anymore.
Yeah, I'm totally not hip and have no idea what that's about.
Although, how many parents are currently monitoring a remotely operated vehicle 2,000 meters down in the Gulf of California? You must be the coolest mother out there.
I think I'm pretty cool. I don't know if my kids think that though. I think they do, but they would rather that I not be away so much. But then again, you know, how many? I don't think many moms get to drive ROV dives.
Yeah.
So that's a pretty cool thing to be able
They're
to
like,
do.
Mom, we don't want to hear about Veggiatoa anymore.
Yeah, exactly. Can you please not talk about tube worms for just a minute? Yeah.
That looks okay. Yeah, I've seen it.
I cannot wait to figure out what these are, these things. I mean, but what's really tragic to me is that you've got these amazing structures, these corals that provide this three-dimensionality for the shrimp to inhabit, and yet the beauty and majesty of it is just, you know, you've all this nasty trash everywhere.
Do you think it compares to like when you get a travel brochure and you're like, oh, I'm going to go to this amazing
Yeah,
exotic
yeah.
destination? You get there and the whole place is
Yeah,
surrounded
the
by
beaches,
building site
yeah,
and the plastic.
yeah.
And,
Yeah. They've taken a picture of the one angle that sort of shows something beautiful, and then you look at the other angles and it's all kind of ugh.
So to take it off on an angle, what would your TripAdvisor review of this area be?
This is a spectacular place, but you know, you're not getting a total escape. You're basically not immune to human impact here. You would think that 2,000 meters below the surface in this incredible place, you would just see beautiful geological and biological features, but you wouldn't see anything else.
And how do we fix this?
Well, we fix it by making people aware that every action matters, and that, you know, nothing goes over the side and just disappears. It all ends up somewhere. And even if you think it, you know, it ends up in places like this where you wouldn't expect it to end up. So I think that's something that everybody can take notice of.
So do you think, I mean, people ask us to collect this stuff, but do you think the biggest aspect is to educate? The biggest weapon you've got here is the 4K cameras.
Yeah, absolutely. I think people will be stunned when they see this footage and realize how much stuff is on the bottom. And, you know, hopefully that'll make them think twice before, you know, and maybe they'll say something when they see somebody littering. They'll say, hey, you know where that's going to end up? It's going to end up potentially on the bottom of the ocean somewhere.
And in your short career, should I say, not of working at sea, have you seen a large increase in the amount of trash in the oceans?
You know, I was just talking, I think, to Tom about this morning. Yeah, when I first started diving, doing work on the seabed, we're going to keep heading. We'll do one little roundabout here and then head towards Falco's Fountain. When I started doing this kind of work 20 years ago, you know, we would see trash occasionally, but it was usually big things like, astonishingly, I saw once a chest freezer on the seabed, and I've seen a number of lawn chairs and other kinds of, you know, furniture on the seabed. But now we see just plastic. We see plastic bags. We see garbage bags. We see various other kinds of plastic debris. It is insidious. It is everywhere. There is no place that I have been working in the deep sea in the last, you know, 5 to 10 years where we didn't just see one bit of garbage. We saw just junk after junk after junk after junk after junk after junk.
I remember reading a thing recently and put it into context. When I first went to sea, which is only 20 years ago, a lot of the people were saying the plastics we saw in the ocean then were mostly products of industrial waste. But now it's more consumer. We're seeing
it's all consumer
microplastics
waste.
from jumpers. We're seeing plastic bags. It's more
I mean, just
everyday people.
Look at all the, I mean, this what we should be looking at right here are these, whatever these corals are with the shrimp and the squat lobsters, and it should just be this sort of landscape peppered with these deep sea tumbleweeds and clams and shrimp, and instead what your eye is drawn to is the trash.
Look at that big piece over there.
You just see the trash.
And will these just, I mean, these will never go away. They'll just break down into smaller and smaller pieces, right?
Exactly. exactly.
So,
And there's a squid that's caught in it. See that? Hold stop for a second.
is that a squid?
I think.
Uh,
Oh, I can't see it.
You let me back up. Just keep
Yeah.
it balanced.
something in there. I don't know what it is. But yeah, the sheer amount of plastics there is mind-blowing.
So my daughter was just listening, and she agrees that I have a cool job.
Ah,
So-
excellent. So,
But she also agrees that I'm gone too much. So
Okay.
she said yes to both. But I think she's okay with the cool job part.
I think the cool job
It's
thing.
bragging rights.
I
You're
think.
friends, right?
Oh, let's zoom out. Should we? Let's see. I think sometimes to go and do this sort of science, there is sacrifices to be made.
Absolutely.
Unfortunately.
And I don't think people realize that, you know, it's very difficult to be away. I mean, you guys were what, on 2-month stints?
And I think that modern society, because we're so interconnected with social media and stuff, people, like, you know, everyone instant gratification, you know, for families at home, like, people just find it mind-blowing that you can be away for 2 months. And, you know, it's great. We've got on board Falco amazing communication with our family, but
Yeah.
I think the modern mindset has moved away. And even for us, you think 40 years ago, people would go to sea and they'd only get written letters in port, or
Yeah.
the occasional phone call that got patched through various
Oh, that
countries.
that cost a small fortune per
Yeah.
minute. Yeah.
I mean, look at this trench at the moment.
Oh my
the
gosh,
Somme
guys, look
battle.
at this.
Let's
This
get
is
some imagery of that.
this
We get
is-
highlights, John.
yeah, please. This is low
Well,
lights.
Yeah,
but
this isn't video gold. This is video. I don't know. Top.
depressing.
Yeah.
Yeah. Video black. So this is a naturally occurring fault line that we're going down, and you can just see with your own eyes the accumulation of garbage in this trench. Now, what you see that looks like the wiry structures, those are naturally occurring. Those are supposed to be there. All the rest of it, the plastic bags, we're going to go back through this video and count the plastic bags and post a number for you all to see. But the number of garbage bags is just, it's ridiculous. It's embarrassing that it's unacceptable that we're looking here. Here we come to the field of there's the frozen balloons.
Oh, let's have a look at some of these balloons. I wonder where they came
Yeah,
from.
let's look at those.
And don't worry, folks, we're going to move you away from the depressing stuff.
Yeah, in
soon.
just a minute. We might need to turn around in order to see it, though. So here, I'm going to zoom in here. These are balloons from, you know, like birthday parties that, you know, somebody probably released in joy after their birthday party and we're like, woo, I'm going to watch the balloon float away. Sadly, that balloon came down here and landed on the bottom of the ocean in this trench. We're going to turn around and try to see if we can see anything on these balloons. This is the frozen spot. I think this is the place where we saw it.
So yeah, folks, we'll be. I know it's depressing to see, and a lot of you came here for the cool imagery. We are transiting from one amazing vent field to the other, but along the way, we're just taking in some of this trash, basically, that we're seeing along the way. And this is equally important. Communicating the amazingness of these hydrothermal vents is our main objective during this mission. Although equally is communicating all this junk that we are seeing deep in the ocean. We're not, some people are asking, are we like Greenpeace or that? No, we're just
We're doing
normal
science.
We're doing science, but it's hard not to see the impact that this is being caused and take something away from this. We're not extreme in our science, we're just mind blown by what we're seeing here.
Yeah, let's keep going. Let's keep going. Yeah.
Okay, guys, we're gonna head on
headed
over
towards Falco's
Fountain now, which is an amazingly picturesque underwater hydrothermal vent.
We're about 5 minutes, 10 minutes out from that site. So if you've got any questions, please shoot them our way.
And some people asking, who are we? So this is the images are coming to you from remotely operated vehicle SuBastian. Now this is operated off research vessel Falco, which is owned and operated by Schmidt Ocean Institute. And you can visit our website, www.schmidtocean.org. And you can also find us on Facebook, and you can also find us on YouTube. There's a squid down there. Is that all mangled? And there's a balloon next to it. So this, the reason we're also running along this fracture is it takes us from Alvin Spire all the way to Falco's fountain. As Professor Joy was saying, Falco's fountain was named by one of our viewers the other day. Hopefully at the end of this dive we'll be visiting some other places that haven't been explored, and usually we put out the opportunity to name these features to those watching at home.
So we're still moving along this fault. How much further are we? We're about halfway there. We're about halfway. No, we're a little bit closer.
No, not much further.
10 minutes, yeah, 10 minutes max. But you can see this depression in the seabed as we're moving along an active fault line. This area is impacted by hydrothermalism. It's really active. There's a lot of flow features. Some that are, the pagoda structures are generated in really high flow areas. Other areas like the microbial mats, which you'll see as we get closer to Falco's fountain, are driven by more diffuse flow regimes. But all of these features are just, they're related and interrelated and connected to the broader sort of geologic setting in this region, which is, accelerated by geochemical dynamics because of hydrothermalism.
So John is just showing where we are along this fracture feature on the 3D image, picture-in-picture that you're seeing on the live feed. So in the distance, I believe that's Falco's fountain, right, John? Yep, John gives a nod. And we're heading over there now. So the little yellow square you see is a 3D representation of Sebastian. And that is where we're heading. So you can see the trench fault feature to our right.
Yep.
And we should be there in roughly 10 minutes or so. So stay tuned. There's a little bit of bacterial mat or something on the seabed there. And you can really start to get a feel for why these are called oases.
Yeah, because you go from just sort of general background sediment, which is brown and somewhat boring looking, to just phenomenal accumulations of biomass and diversity over a very short spatial distance.
So while we're transiting, we can see that there's a lot of people online. Ask your questions, ask away, and we'll do our best to answer them in the control room. Currently got Professor Joy, ROV team. What have we got here? Is it another balloon?
Balloons.
More balloons.
God.
Looks like another balloon.
Yep. And there's another one coming up too.
So you're seeing here is another fracture as we make our way to Falco's fountain, which is accumulation of rubbish. That really gives a good overview of the fracture.
Yeah. There's a natural and unnatural. It's a great view of the fault though. You can see how deep it is.
There's a big hole there. I wonder what lives in that.
Oh, there's more. There's anemones down there. There's some,
It's amazingly straight there.
yeah. Looks like somebody chiseled it out, doesn't it?
What are the main difficulties of working at this depth?
The main difficulties, well, I'd say definitely the technology, and hopefully we'll get Russ, our ROV supervisor, on in a bit to talk about the challenges with driving a vehicle down here. I think it's put the science, I mean.
There's another trash bag.
Oh, sorry, we're just looking at something.
Go ahead, Bridge. I do, yes. I'm gonna keep moving down a little bit more, so I'll just slightly follow it down.
So I mean, at the moment, Tom, the vehicle's being subjected to about
Sway a little
2,960
bit to port.
pounds per square inch of pressure.
Leighton.
Oh, what have we got here? Is that Doritos?
I think it is
No, it's plastic.
Cool Ranch Doritos and another balloon.
Aye, someone's been having a party.
Party, yeah.
Aye.
And I think that is just netting.
Is that the end of the fracture, though?
Nope. Well, it might be the end of the expression, but I think it actually splits into a wider feature. I mean, we tracked it all the way, I
Yeah.
believe, to Falco's Fountain.
Someone's asking if we've seen any sharks here. Do you want to explain about the oxygen levels that we're seeing here? Then?
Yeah, so one of the really unique things about the Guaymas Basin and one of the fascinating aspects for me as a microbiologist is that in most hydrothermal systems, oxygen concentrations in the bottom water are quite high. They're, a couple of hundred micromolar, which is, a good deal of oxygen. Here in the Guaymas Basin, the bottom waters are what we call hypoxic. So the oxygen concentrations are 33 micromoles per liter. So about a tenth of what you get in, active surface water, and much, much, much lower than you would expect to have in cold, deep water. So what that does in principle is it limits the types of higher animals that you get here. We're seeing a lot of fish and squat lobster and shrimp and octopus here in these places where we're tracking. But what you don't see is a lot of sharks and a lot of big squid and a lot of the more sort of charismatic deep water fauna that you would expect to see. And I don't mean to infer that Riftia isn't charismatic, but they're sessile. They're not moving around and poking the vehicle and giving us the side eye the way that big sharks do. But the reason we don't get those big animals is because they would literally have a problem breathing down here because the oxygen concentrations are so low. So we continue to see trash on the bottom in this topographic low. We're beginning to sort of pick up some hints of activity along this fault. The black reducing sediments are sulfide staining of the sediments, which are highlights of discharge. And up on the right hand top of the screen you see some little spires that are a little field of spires. This is a hot spot of activity that we've sampled before. These are all areas of diffuse flow, active discharge, and there you see the orange anemones and bacterial mats, and of course it wouldn't be the same without seeing a big piece of trash right there in the middle, a big plastic bag, all right, in the middle of the field. And we didn't plan to show you guys. We picked this path. It's the closest way to get from point A to point B, but it's also an area that is just incredibly impacted by trash. This is not unusual, though. We've seen this on every dive.
Is that tube worm or is it plastic again?
I think that's a tube worm right there, but that's plastic on the bottom. And here's there's an octopus. And then we've got, so all of these mineral pavements that you see right here, these are evidence of actively venting fluids and water rock reactions.
So how come some of these, we saw those...
And we're just heading towards the next pagoda to show you all. But yeah, this is par for the course as we've seen during this expedition. There's just, there's trash everywhere that you look.
So are these faults caused by earthquakes?
They're caused by tectonic activity. So yeah, natural I'm sure there was an earthquake when it formed. That pull apart trenches. Here's another one. Looks like a cat. Is that a balloon or a piñata or what? Trash bag. There's another trash bag.
Plastic bag fish.
Yeah. Here's a bucket. We haven't seen a paint bucket yet. Here's a paint bucket. Wow.
Sorry, Tom, it's not as if we can make Our outreach guy wants us to make it like picturesque.
We're almost there.
We're there,
We're
Tom.
almost.
Not in the slightest.
No, I'm only joking, Tom.
Oh God, this is crazy. Is that a coconut?
Sorry.
Is that a coconut? What is that? Okay, we don't have to stay here. We should go. I'm depressed. I want to see something beautiful. What? There's a big anemone. That's a big anemone. Whoa. We diverted there.
Can you explain why these are more orange here then?
So the orange is potentially freshly precipitated metal oxides. It's really hard to say without sampling them, though. When you get around areas of active discharge, some of the mineral colors are quite bright. You can get purples and greens and oranges. Some of the exotic metal sulfides are orange, which is mercury and arsenic in particular. It can be orangey looking. So there's some really, you can't define the mineral by the color, but you can sort of get a good guess of what it might be from the color. Oh, here comes the microbial mat. Here we go. We're pretty close. Here's a chimney field. So you can see there's small chimney fields all around the edge of this fault line. No, we're not there yet. Yeah. But we're gonna keep going to Falco's Fountain. Oh, there's some diffuse flow right there. If you keep your eyes peeled, you'll see shimmering water, and that's a bullseye for where the diffuse flow field is. But we are gonna sort of move a little bit to the south along this line. There's a jellyfish, our first of the day. Oh, that's sulfur. Yeah. It's dead ahead. Yeah. To the port, yeah. So at the bottom of these features, we see these boulders. These boulders are probably relic flanges that became unstable and just came crashing down. As we move closer, you'll see a lot of chemosynthetic tube worms, riftia. This area is quite interesting. You're going to see a lot of blue and purple here. It's quite spectacular in terms of the colors that we see, and that's telling us something. We haven't quite figured it out. The deep sea provides us with a code that we have to decipher. And the colors, the chemistry, it's all telling us something about how this feature works and what makes it tick. And our job as biogeochemists and microbiologists is to take all of this information and distill it and determine how it all works. Yeah. Come out.
No, just
Yeah. 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 I should let you run the camera.
Can you talk a bit about chemosynthesis in general? If people
Sure.
don't know much about,
Yeah.
how does life survive when there's no light?
So chemosynthesis is a type of autotrophic growth, and autotrophic means that you fix carbon dioxide into your biomass. So instead of, like, we are heterotrophs, we have to consume things like burgers and pasta and bread and whatnot to get our energy. These animals get their structural carbon and from CO2, and they get their energy from chemosynthesis. So this is Falco's Fountain. Falco's Fountain, as you'll see in a moment, is a really spectacular feature. There's flow under these flanges, and they are just incredibly complex geologic structures. And we found this place a couple of days ago, last Thursday, I believe, and we were just absolutely mystified by it. At first, we thought this was a dead chimney, and then we realized, nope, It's not a dead chimney. It's flowing. And if you look right there, you'll see the interface. Can you go up a little bit more? You'll see that interface come into view as the light changes from, we can pierce. Let's take a view in there and see if that's the same. See, these are different from what we saw before. These are minerals that are forming inside the fluid. And look at all the amazing colors here. Good God. This is crazy. I just cannot wrap my head around it still. Here we go. Here comes the shift. Ring. We gotta wait for it to clear up a little bit. It'll shift when you go up. Bing. There it is. Oh, we're getting yanked. Is this ship behind us?
Just the way he's moving.
Yeah. So we're having some, we have to wait for our tether to get into the right position. But you can see down in the lower part of your image these beautiful purples and reds beside the riftia. There's anemones, all kinds of animals and microorganisms living on these structures. The colorful mats. Yeah.
So where does all the material that builds these chimneys actually come from? How does it get there?
So the hydrothermal fluid is loaded with reduced materials, so building blocks for these structures. And when that fluid interacts with the seawater, you get precipitation of minerals and chimney structures. We can estimate, once we get the chemistry of the fluid nailed down, how long, we know that it has X amount of these materials in it. You can sort of you can estimate what the flow would have to be to generate a structure like this, which is an interesting calculation to make. Yeah. Yeah. All right, so we're gonna motor up and down a couple times here. We're gonna collect a water sample. We're probably gonna collect some more of these microbial mat samples at this site. We'll be here for probably about 45 minutes, sampling and documenting the structure. It's one of the most beautiful pagoda structures that we've visited on this expedition. I just love the purple. It's just purple's my favorite color anyway, but this purple is pretty spectacular.
It's
hydrothermal purple.
Is that
yeah.
your new bathroom
That's
color?
my new bathroom color. I'm gonna have I think I might have a problem. I might have to get somebody to make it for me, but yeah, hydrothermal purple is my new favorite color. That's
Color in the paint shop.
yeah. Wow, look at that. I didn't see that before. This thing right here. It's not focusing. It's too much turbidity. Okay, we're gonna give you a flyby of this feature in a second. We're gonna go up and down and you can see the interface. Here we go. Ring, disappears, reappears. It's hard to see. That's not the big chimney we saw the other day. That was shimmering. Can we go around and find that one that's got the massive flow? But look at all these. I mean, the riftia bushes here are phenomenal.
So the way the riftia make a living is pretty unusual.
Yes, these organisms are chemoautotrophic. They don't have a mouth, a gut, or an anus. They have instead an internal compartment called a trophosome where they farm chemosynthetic bacteria. And these bacteria provide them with their nutrition. And you can see among these riftia beds, these lush orange and white microbial mats are also chemoautotrophic. And chemosynthesis is a Oh my goodness. Chemosynthesis is a naturally occurring process that happens in places where there is no sunlight, and in such places the organisms use the energy from these geothermal fluids along with electron acceptors that are in the seawater to make a living. So they carry out what we call chemosynthesis. It's very similar to photosynthesis. It's converting chemical energy into metabolic energy, and they use that energy to proliferate and to accumulate, as you can see, extensive biomass in the environment. There are Riftia tube worms in this picture. There are para-alvinellids, the little guys with tentacles. There are some alvinellids in this picture. There are cnidarians. There are tube anemones. There are a number of different microbial features, and then this amazing mirror flange. This is kind of like an underwater, upside down waterfall, where the hydrothermal fluids go up because they're more buoyant once they've been released from the edge of the chimney. These are just jaw-dropping, stunning features to see. We're so happy to be able to share these images with you because they're otherworldly. You just don't. It's something else to be able to be a part of a discovery of something like this. Wow.
So it's pretty hot in there, right? Do you know?
It's about, this one was around 270 degrees centigrade.
So that would be a bit too hot for us, right?
Just a wee bit.
But there's like viruses and microbes.
There's viruses and microbes that are growing right on the edge of the chimney, and there are organisms living in the rock that the fluids are flowing through. This is a living structure. It's not an inert rock. We're really interested in understanding the interaction of biology and chemistry here. Look at that. And it's awe-inspiring. The fact that you can see the reflection of the biological communities in the rock, or on the interface of the bottom side of the flange is just, I still am just stunned seeing these beautiful structures.
Can we do a little flyby to see the change in angles? Is that possible?
Yeah.
I think we should get it.
So the riftia there that we can see through the shimmering water.
Yeah, they're living at the edge.
They don't want to be right in there.
They can't be right in there because they would essentially be boiled. But they're living right on the edge of what's tolerable for them, and it's pretty incredible that I think they're being very adventurous, these riftia, because they're just right on the backside of what's would be tolerable for their metabolism.
So if you look at it now, it looks as if there's nothing there, right?
Yep, looks empty, and then kaboom, there's the reflection.
And that's because
The ocean, looking at it through an ROV eyes instead of the side of a small fishing boat. This is something, because that just looks like some sort of a black cavern, right? Now it looks empty. You can see you sort of see these minerals sparkling. If I zoom in on that, you'll see the minerals kind of dancing behind. And then occasionally you get this little hint that there's an interface there. And then as we come up, you'll see the interface reflect. This is outrageous.
Some people are saying it's like another world there, and it's kind of interesting that people think this type of way to make a living, this chemosynthetic life, is something that could exist on other planets.
Absolutely. I mean, these extreme environments have a lot to teach us about life on our own world, as well as life that potentially exists on, other planets and planetary bodies. Conditions that are challenging for life on Earth provide an opportunity to learn about life's flexibility and pliability and adaptability. And you look at these structures and you're surrounded by the awe of microbiology and the ability of these organisms to just take advantage of every type of chemical disequilibria to thrive. I mean, look at these phenomenal, I mean, these are just like, you know, we were joking the other day that they look like thing 1 and thing 2 in caverns, you know, that stringy hair. These are all Beggiatoa filaments, we think. They look like it under the microscope. We're going to collect some more of this material today, but these stringers, these are not in super hot fluids right now. This is a pretty cold side of the chimney, but it's still impacted by the discharge of this energy-rich fluid, and there's plenty of metabolic juice to keep these organisms growing and happy. We don't know what the purplish colored mat is, but the pink and the orange and the white, at least the stringy spaghetti white, are different flavors of Beggiatoa. Then you've got these white elemental sulfury coatings that tend to be dominated by other organisms. Here you can see the source of diffuse flow feeding this particular part of the chimney. It's right down here.
People are asking how big is that interface? They're getting a bit lost with the sense of scale. Can you give us a bit of an idea of how big this whole system is?
That flange is what, 2 meters across? So it's a fairly large frame that you're looking at right there. You're not close up. We're gonna sit down and sample. I want to sample the this stuff right here. This white stringy stuff, and I want to get more of this pink stuff around the paraalbinellids. We're gonna do 3 slurps here: white, pink, and orange. If you can, we're gonna spend a few minutes collecting some of these microbial mats. We're really interested in the natural products of these microorganisms, particularly their sugars and liposugars, the ways that they basically use to talk to each other, the signaling mechanisms that they use. We're going to be doing some metabolomics work in addition to metagenomics. Those 2 green lasers that you see are 10 centimeters apart. So that gives you a feel for the scale of this feature.
Where do you see it? We can put our eyes in there.
They're in the back of the cave. Yeah, yeah.
Oh, I see mine.
Trying to find, instead of typing every time I copy and paste when I don't screw up. Boop, 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 boop. Wow, look at that shot. Oh my gosh. Whoa. Yeah, something is unique about those white stringy. And see, this is the other, I don't know what this is. This is, yeah, we're gonna.
Can you talk a bit about the sort of microbial architecture? They seem to be building quite a range of different shapes.
Sure.
Can you talk us through that?
Sure. So when you see the surfaces, they're coated with these mats, but you'll notice that it's not just a smooth coating. It's very rough and almost purposeful. It has holes and caverns in it, and topographic highs and topographic lows. Well, what the organisms are doing, they generate an architecture. They're essentially environmental engineers, and by generating architecture and complexity, it accelerates and enhances fluid flow across the structure. So it allows them to accelerate substrate delivery of things like oxygen and sulfide to stimulate their metabolism, which is really just amazing when you think about the fact that, you know, these are microorganisms. They don't have brains the way that we think about brains, but yet they assemble and generate complex architecture that affects their surroundings and affects their well-being. So they're generally building a structure that helps them be more successful, even though they don't have brains. They're doing that on purpose, which is interesting to think about.
So you talk us through how you go about sampling those, you know.
So there's a couple of things that we can do to sample them. We can put out settling blocks and you know, see how things settle and grow over short periods of time, or we can just use sediment cores to sample them. We can't do that obviously on this rock, so we use what's called a slurp. And the slurp gun is basically a big suction hose, and it allows us, we can point to a part of the rock and ask the pilot to please suction this particular sample into this particular rock, into this particular tube. So
how do you choose a site? What's getting you interested in saying I want to get a sample? They might react to the slurp gun coming on. What would they likely do?
They have brains, and these tube worms have brains, and they're very sensitive, and they will potentially retract into their tubes once they get bothered by the slurp gun, because they want to stay safe from predation.
So crabs might come along and snip a little bit of the plume
Yeah,
off.
they graze on the plume.
But not many crabs would get this far up, right? They don't want to go where it's that hot.
Oh yeah, the crabs are a little wimpy when it comes to temperature. But the Riftia here, you know, we don't see a lot of crabs. I mean, there's a few over here, these squall lobsters, but we haven't seen any of the big spider crabs.
Like coral reefs, they provide this kind of structure, and you can see now in this shot how many other animals are living on, in, around, between. It's a lot easier to make a living if you can hide somewhere occasionally, get some shelter, get into the way of some nutrients, get out of the way of some predators.
Yeah, and the surfaces create habitat for microorganisms, which provide food for these animals. So they basically, you know, they're the way that organisms go onto coral reefs to sort of forage, and you know, they're mobile and they move in and out of the reefs to get their food. The same thing happens with these oases in the deep sea. Animals come in and out to get their foraging. Oh.
Getting good sample in there, number one.
Yep. Oh yeah, that's good. Let's get a little bit more if we can of the pink, and then we'll switch to the why is this not updating? That's a long time ago, 1634. Are we on auto? Are we doing auto? Yeah, we are. Okay, it just hadn't updated. Okay. Yeah. I wanted to get this image so that I could get my not mess my notes up. I'm taking notes of sampling as I talk, and if I'm not really careful, I get confused. So I gotta make sure to write things down.
So what type of analysis are you gonna do on this microbial map when you get it back into the lab?
So we're going to do metagenomic sequencing, which will give us a fingerprint of the microbial community, and then we can assemble we can do this procedure called assembling metagenome assembled genomes. So we can pull genomes of individual organisms out of this larger metagenome. So we can say, you know, pull the pink Veggiatoa out. We can also look for new organisms by developing these metagenome assembled genomes. So we're going to be doing some really complex bioinformatics with these samples that allow us to both fingerprint them in terms of population diversity and dynamic, but also potentially discover some new organisms. Okay, so that's, yeah, let's go flush it, and then we'll go to the white or the orange, whichever.
Okay, awesome.
Yeah, we're having, we're just doing some sampling here.
It's because it's strange. It's really bad.
Someone on the IFLS Facebook group is asking if there's any jobs going, and they're jealous of your job here.
I can do that one.
I want to retire.
You want to retire?
No, not yet. Another 10 years, then you'll be good.
Okay, so get training. How long does it take to train?
It depends what you want to do. I mean, there's plenty of jobs. So on board the ship, not only do we have our scientists, and Professor Joy will speak about like how to become a scientist in a minute. We have people from various engineering backgrounds, technical backgrounds, such as computer science. So yeah, I mean, you can come to oceanography not just through like marine biology or the usual ones, but pilot vehicles like this is, I think, apprenticeship in engineering, some people in our team have a background from experience they gained in the military. And others, we also have a graduate program where we take students who are at university and
Let's
we
try to
bring them
get
to sea and see if they like it.
this
So
stuff.
keep a look on the
Yeah,
website.
I
You
got
know,
you.
jobs are always coming up. Sometimes we're looking for techs, sometimes we're looking for deckhands. You never know.
What
No.
sort of characteristics do you need to make it in the ocean?
You need one of the main characteristics is that you either need to be single, have an understanding family. You are away for a long period of time. We won't lie there. I mean, it is cool to see all these sites, but you may be away for a month, 2 months, 3 months. You know, that's not to take away. Someone was saying, military families sometimes they're away 6 months, a year at a time. It's nothing to take away from their sacrifices they make with their families and stuff, but it is a long time, especially in modern times where people are used to having people around, social media and stuff. So, you know, you need an understanding family or partner. But one of the main characteristics is being able to get on with people. You are in close confinement with people for a long period of time, so you need to get on with people. At the moment, we've just landed. They're going to do some more sampling shortly.
I think that curiosity is the main connecting characteristic of everyone I meet that works on ships. They seem to be, especially in like a science research vessel like RV Falco, whatever job you've got, people are curious about the research that's happening and how they can help make it more productive.
I would agree with that. Curiosity is absolutely essential, and I think the most number two is just work ethic. Working at sea is not easy. It's not a 9-to-5 job. Sometimes it isn't even a 9 to midnight job. Sometimes it's a 24/7 job. But it's the reward, I think, more than makes up for the sacrifice that you make in terms of the effort you put into working at sea. You get to see things and do things and experience things that no one else gets to do. And it's a really special job to have, and I think it's just a privilege to be able to do the kind of work that we do.
Do you get a buzz when you see your science like suddenly on the news or in a paper? And
Absolutely.
you're like, oh wow.
Yeah, I mean, and that it's more of a thrill being able to share it with people.
Yeah.
It's not so much about we're in the news. It's wow, people are going to learn about this and just want to know more. Let's go to this guy right here.
I meant more in the realization that you're so ingrained in it.
We can do this stuff separate if you want. We can do whichever one. We want to get them both. These white fibrous tendrils I'm really jazzed about.
I was more in the sense of like you sometimes lose the picture and then suddenly it's in the news and you realize, wow, taking a step back, this is cool. Like you've probably seen vents in most of the world's oceans, and then suddenly someone's seeing this for the first time.
Yeah, it's really incredible to be able to share this in real time with people as we make the discoveries, and that didn't happen even 10 years ago, right? Or even 2 years ago, really. The ability to live stream dives and whatnot has just improved remarkably over the past few years, and
You can look lazy. It looks a bit like smoke going in there.
it does.
So there's a really interesting strategy they've got to kind of capture food in this environment.
High surface area to volume and really flexible. They can move with the water. And they're kind of difficult to sample because they clearly have a good strategy. The tensile strength of those filaments is probably pretty good.
Yeah.
Yeah. But see, that means they have really interesting goopy structures that are holding them on the rock, and that's why I want them all. If we can, yeah.
So most people won't get the chance to ever come and see a hydrothermal vent. So what is the closest thing on land that they could go and see?
Yellowstone
Yellowstone.
National Park.
Never been myself.
Oh, no way.
I know.
Oh, shame. And the hot springs in California along the Long Valley Caldera are pretty spectacular too.
Would they be similar to the sort of things you've seen in Iceland then?
Oh yeah, Iceland is
like geothermal.
Yeah,
pools
I mean, I've visited a lot of the world's geothermal environments on land, and I think Yellowstone's by far my favorite. Iceland is, Iceland is pretty
phenomenal.
amazing. There's some incredible places in China too, but Yellowstone is just remarkable. I mean, it's a lot like these deep sea environments. You can go high pH, low pH, super hot, super cold. prismatic is one of
Yeah.
just one of the most jaw-dropping things imaginable.
Do you see any similarities between the ecosystems there and what you're seeing deep under the ocean?
Yeah. I mean, the fact that you have, similar clades of hyperthermophiles, or types of metabolisms, at least, is quite fascinating. I think there's a lot that we will learn from these organisms that we're looking at right now in terms of novel metabolic capabilities and strategies that they use to be successful. Like, I'm super interested in how are they glued to the rock?
Yeah.
What chemicals are helping them stay glued to that rock and stay so stable in the system, and we can determine that by doing metabolomics and looking particularly at their carbohydrates. So we're really excited to be able to do that, and looking forward to carrying out that work.
Is it really hard getting these samples to the surface? I mean, as opposed to, do they get denatured or in a way that makes them hard to study?
So they actually do pretty well. They're not piezophilic, so they don't require high pressure. And the slurp they're not so concentrated that they go into shock or competition overload. We feel, obviously doing like an ice smash sample and pickling them at the seafloor for their RNA and DNA would be good to do. But we're not doing that on this trip. We're planning to do that the next time we come here. This is just a grand overview to get a lot of samples for doing an initial first pass, generating data for future proposals so that we can come back and do more. Of course, publish.
To jump back to the, say, Yellowstone and the other vent systems in, say, Iceland, we know that there's hydrothermal vents all over the ocean, and some of the questions are the similarities between them. How can they be similar if you can't survive off vent? How do they populate from one vent to the other, and how are there similarities?
That's a great question, and it's one that I think is fair to say baffles people still. One way that we think this happens is that there are stepping stones or hot spots, because these organisms can't survive long distances off the vent. So we think that things like whale falls serve as sort of a stepping stone for these populations to come into different environments and sort of radiate across the deep sea.
And a lot of people are asking, what plate are we currently on and to have all this activity?
So the North American plate is to our east, and the Pacific plate is to our west, and the Pacific plate is moving to the southeast, and the North American plate is moving to the northwest. And as they move past each other, this transform fault is generating.
So is this spreading or
this
friction?
It's spreading and friction. So this is the East Pacific Rise.
Okay.
And when the East Pacific Rise hits North America, it turns into the San Andreas Fault, which most people have heard about because California earthquakes, Mexico earthquakes,
And the rock.
Yes. So are we clogged, or are we just?
We got a lot of the surface stuff off now.
So what you're seeing them using at the moment is their underwater suction sampler. It's kind of like a vacuum cleaner, and we're just sucking some of this bacterial mat off the rocks. And Professor Joy and her team will study this once it's brought on board.
Yeah, there's quite a bit in there,
But it just hasn't come through to the
It's coming
jar.
through. It's just,
Okay.
It does clog quite quickly. So,
Yeah.
Wow, someone's asking, what is the usual lifespan of a vent? I think that's a piece of string question.
I don't know. We're gonna try and figure that out. We know that these structures changed rapidly because we visited one of them in November, and it was really different compared to now. Wait. I don't know if that's filamentous. Is that filamentous? I can't see. We don't want to get the jello in there with the filaments. Yeah, I would stay over there if you can, because there's jello over there too, and the jello is a different beast. Is that our max reach?
Yeah.
Well, let's see if we can shake the arm and get the rest of the material discharged. Oh, that was a good one. Yeah, you could, right here. Yeah, maybe get this guy right here.
Someone was asking just now, and I'll answer this one. How can they encourage their children to want to do such a cool job? I don't think it needs much encouragement. When you look at the screen, you've got a robot that is diving 2,000 meters down. It's controlled by a team on board. is visiting parts of the planet no one else has ever gone before. This is true exploration.
I think we got enough. Let's
Yeah.
shake it.
A lot of kids these days love technology, love
Yeah.
equipment. I mean, just look at some of the equipment we use in here. I mean, it truly is an amazing job. It needs very little encouragement.
I think so too, and I think
It's
it's really
just the
just making
opportunity.
people aware of it.
Yeah.
It's not on. It's not on.
The you know, I didn't really know that.
You're gonna put.
I had no idea that you could do this kind of work
Yeah.
when I was a kid, and I certainly, you know, I didn't even know in college until I took my first oceanography class.
Alright, we're off. We're off. We're off.
Yeah, pull off.
Okay, one of the difficulties with sampling under the water is like as the manipulators push on stuff, we don't know the consistency of the rocks, and sometimes we can get pushed off. In this case, the ROV's just pushed off the structure just to make sure that we don't damage it. And now we're going to go back in slowly and resample. And just to go back to what Andy was saying there, a lot of people aren't aware of this as a career path, so just encouraging them to look at this. There's some amazing institutes in America and across the world that support this, like Scripps, Woods Hole Oceanographic. Where are you from, Andy?
University of Georgia in
That was
Athens.
The University of Georgia. In the UK, you've got like the National Oceanography Centre. We've also got up in St Andrews, and then SAMS, Scottish. So, if your kids or young adults are interested in oceanography, look it up.
And the ocean is incredibly big, and our opportunities to explore it are relatively small, so you need people who have great ideas for ways we can continually improve technology. A lot of things have changed in recent years that allow us to make more advantage of, you know, we only get to be at sea a certain number of days per year. We can only run so many samples. There's a lot of things that we would like to do that currently there isn't enough time to do. So there's a lot of opportunity for people to come up with creative solutions. And in oceanography, a lot of the time people take an idea or a technology from one setting and they find a way to use it in the ocean. And so it's really great if people who have other backgrounds other than ocean sciences collaborate with people that work in oceanography and can come up with some ideas of how they can work in these environments.
I think, yeah, that's spot on there, Tom. I mean, you look at how oceanography has changed, even in the last 20, 30 years. Remotely operated vehicles, really, they started to appear in the mid-80s. Most people became familiar with them in science after Woods Hole's exploration of the Titanic, but since then they've become quite a commonplace, not that common though, piece of equipment for an oceanographic institute because of the amazing pinpoint sampling we can do here. But at the same time, the technology's exploded. The sonars on the ship, the technologies that make it possible to go down and run SuBastian at this
There's
depth.
some good orange.
So people with a technology background and big ideas are the people that are needed.
Yeah, you can get that up there.
Hello to the people on the Isle of Skye. You're surrounded by the sea.
Yeah, everyone give us a shout out, tell us where you're watching in from today. We're currently in the Gulf of California, off of Mexico. Yeah, where are you?
I was also amazed at chatting with people who live maybe in
Yeah, that
the
orange is
middle of
good right
America,
there.
who don't see the ocean on a daily basis. A lot of us, I don't know about you guys, I grew up living at the close to the sea and seeing it every day. So they were saying, someone I was chatting to was like, this is even more like wow to them, because it's a whole world. They don't know the sea, waking up and seeing it every day, but then seeing something like this is, I don't know, when you live on the prairies maybe or somewhere like that, it's totally different to everyday life. And we take that for granted. It's not.
Well, it's kind of amazing, people at home are seeing this exactly the same way we're seeing it on these screens as the ROV SuBastian is 2,000 meters below us. And yeah, we're all kind of exploring together, seeing some of these things in detail for the first time.
Someone's saying the University of Washington, they're jumping on the thing, has awesome opportunities if you want to go and study the ocean. Check them out.
University of Victoria for Canadian.
Yes, that's correct.
University of
UVic is
Oh.
awesome.
Sciences, yes.
I know, we've got Mexican one here. UNAM.
Okay.
Yes, and the folks at UNAM have asked me to express our gratitude to the ship's crew, the ROV team, and the science staff on board for supporting this excellent work and making this expedition possible. So all the folks in Mexico City are super excited to be a part of this remotely.
I'm blushing.
Carlos, happy birthday.
Well, you made it easy to work with.
Alright, go for it.
One of the other major pluses of supporting science at sea is
Can
the
you
different scientists
stay
you get to
above
work
that
with
line so
all
we
over
don't get the
world
pink
and
in there? Yeah, don't get the pink
different
in there.
applications of science. No 2 days at sea are ever the same. And it can't be said about many jobs where you go into the office, work on a computer, you come out here, the light's different, the people are different, the mission's different.
Something else that's different is the smell, though. I mean, when this
Oh,
stuff.
God, yeah.
I mean,
Hey,
it's
he's had a shower
that
today.
wasn't to do with the crew. Sorry.
Speak for yourself.
I'm talking about the microbial mats and the mud cores and the water samples. There is a noticeable smell down here, isn't there?
It
So you don't get in a normal
It's-
office.
It's iodisulfide.
Unless you live
Yeah.
in a refinery, I guess.
Yeah.
Well, someone said it was like changing a diaper in a gas station was the smell of the samples when they came back up.
Changing a diaper in a gas station
When you fed your kid eggs for the
when you
last
fed your
month.
kid rotten eggs for a month. Yeah.
Wow.
Yeah.
See, that's how to get. Yeah, that's that tells you there's something interesting down there, right?
Can you get a
Someone's
little
asking
bit of
if
this
we've got
stuff?
any Australians. Yes, we do. We have a
Say
few
again?
I
Australians.
gotta figure out how to approach it to them over
Okay. Yep. Gotcha.
Some.
No.
I can't get started on that.
Heavy is for Turkians.
So keep your questions coming. We've got a whole team here at the moment. We're just sucking some of this bacterial map. This will be brought up and studied by Professor Joy's team on board the ship. At the moment, these images are coming to you from remotely operated vehicle SuBastian, which is currently 2,015 meters below the Gulf of California, studying some of the world's most picturesque and fantastic hydrothermal vents.
The stuff chokes real quick.
Yeah.
The arm that you're seeing there is being controlled by one of our team in the control room. He's using an exact replica of the arm you see there. Every time he moves one of the parts of the arm on the controller up here, the arm on SuBastian mimics that movement. It's not as easy as those grab a bear fairground attractions where you're using a claw.
Someone on the feed is asking if the hose comes with a scrub brush attachment.
No, the scrub. Yeah.
Push on there.
Yeah. Yeah.
You've got to be creative down here, haven't you? You can come across something that you've never seen before, and then the guys on board will have to work out. Okay, let's go back down there and try and, when we're back on the ship, try and work out a way to change our sampling equipment to get this thing we've never seen before. And
Yeah. I think we got it. I think we got enough. Let's let it suction through.
Russ on board has a 3D printer, which we've used to print new.
One is the white yellow material.
In a moment, we'll show you the manipulator arms being
We're going
used.
to move from this location.
Yeah, that's all right.
to the new one.
And the other thing that we want to do here is collect a water sample at the edge of the flange as we're
Just
moving out.
relocating the vehicle to continue sampling. So on your live feed now, you'll be able to see co-pilot. He's got the manipulator controls there. In a moment, we'll be using them and you'll be able to see. This is what's called the master controller. As he moves the section of that arm, the section on SuBastian mimics that movement. One more.
That's not lightage.
Someone's asking if we've tried turning the matrix on and off again, and it has felt a bit like that at times on this cruise, hasn't it?
It has, and
We've-
we will dazzle the matrix in a few minutes. But yeah, it's really like going into another dimension when you're moving up and down across these chimneys.
Half the
It's
time you want
amazing.
Hello, these live video feed is coming from Europa. No, it's actually coming from the Gulf of California. Feels like we're on another planet.
Do you
at
think
times.
we could sit? Yeah. You read my mind. Because this just looks, yeah. What? Oh my goodness, this is ridiculous. Here we have yellow and orange.
So has there been any discoveries on vents that have helped, like the medical community or anything that's come out?
There are antibiotics that have been discovered from vent critters. I believe there is a anti-cancer drug that was pulled out of a hydrothermal microorganism. You know, these environments have not received the attention of, you know, coral reefs in terms of natural products chemistry. It's something that's fairly new. So I think this is it's a new field that's ripe with opportunity.
There's a cool technical question that just came in. Does the ROV have an auto hover function?
Yes.
Yes,
It does.
It can hover. It can auto hover. We can do auto depth or auto altitude off the seabed. We can also lock its XYZ position, and it'll use its thrusters and try and keep there. Auto heading, so we can say we want to stay on this heading. So there's a number of auto features. A lot of the times around these vents, though, they're quite delicate, and our pilots are almost always flying the vehicle on the joystick. So there's always hands on the controls while we're working here. At the moment, we've just moved off the vent. We'll get back to some cool imagery in a moment. Just coming into your view now. Someone says, "Oh, it's so pure. The ocean is so clean here." I guess they were a viewer that wasn't with us about an hour ago when we were looking at all the junk. So if you get a chance,
I think maybe.
we're seeing the things like the rift here. Are they feeding off each other, like the bacteria? Is that eaten by other creatures, or is it just they're all growing and it's just...
The microbial mats are consumed by other critters. Everything is part of the food web here, and ultimately gets, you know, consumed by something. These mats grow and wax and wane, and everything is ultimately, you know, a part of the food web and consumed.
Someone's asking how we prevent cross-contamination from sites here. The vehicle is washed between dives to prevent that, but the vent system we're working around at the moment is pretty geographically small. So I think most of these vents here in this field, they've got the same biology.
They're connected, yeah. There's not... I doubt very seriously that if you notice the water moving past the vehicle, that water is moving as much material across between these venting fissures as we are. Right in here? That might be good. Let me zoom in on it and take it. Yeah, let me look real quick.
Okay, someone's asking, is the ROV connected by a cable? This is a remotely operated vehicle.
Yeah, that's good.
So it is being operated by
that's
a pilot
perfect,
and technician
actually.
who are on board the vessel. It is connected to SuBastian via 7,000 meters of steel armored umbilical, in which there's fiber optics, and we send 4,000 volts down the cable to power the vehicle. So it is permanently connected to the ship. This cable is about 19 millimeters in size. And that's where the term remotely operated comes from. There are other vehicles in the science fleet, such as Alvin, which is a human occupied vehicle, a HOV. There's untethered vehicles like Alvin, and then there's ones which don't have people in, autonomous underwater vehicles. These are basically self-thinking robots. We give them a mission and they go out and execute them. At the end of the mission, they come up to the surface. Someone's asking, are we in the future? We're not technically in the future, and I know you're referring to the date on the display, which most of you are thinking we're on the 3rd of October again. It's the 10th of March, and the time 1749 is in UTC. So if you're wondering why the date looks different, it's year, month, day.
Day, month, year. This is year, month, day.
That's year, month, day. Sorry, I'm looking at a different feed from you, aren't I?
Yeah.
You've got day, month, year on yours. I'm looking at this one, which is stream.
Not staying on down. Can't get it stay down.
On this one, Tom.
The other day, it cost so much.
So at the moment, our pilots are just maneuvering the vehicle. Now we've got to be mindful. Obviously, there's overhangs. There is current here as well. Someone's asking if we are in the future, can we send them the sports details so they can put some bets on? Sorry, it may feel like we're in the future with all this technology.
We are slightly in the future. We're probably a couple of seconds ahead of people at home.
Oh yeah.
So, I don't know. I don't think we can find a way to play the system though.
I think England beat Italy at rugby though. So we've just come off. We're struggling there with the current coming round and the actual terrain. It's hard landed on some of these small outcrops. So there's a great view at the top of the feature here at the moment. You can see one of the small chimneys. This is Falco's fountain. It was discovered a few days ago and named by one of our viewers at home. Someone's asking if this is a hydrothermal vent site, how come the temperature is only 3 degrees? Now the actual water coming out of the vent itself is around 366 degrees Celsius, which is about 660 degrees Fahrenheit. This dissipates really quickly, so around the vent itself it'll be 4 or 5 degrees. The moment you'll see ours is roughly 3 degrees. We don't put the vehicle too close to the vent because some of the stuff on there will melt. So you may see fluctuations around 4 or 5 degrees. But compared to the actual ambient temperature, which is around 2.8, 2.9, this is quite a considerable increase in the deep ocean. So,
They look at the end of the tube of the second cycle, how it's white.
yeah. Our co-pilot's just pointing out, if you look at the suction sample of that hose, the Perspex tube you see, you'll see that it's bleached white. It's because that's been around the 300 degree water, so it's actually changed the color.
And we're also joined now by Jesse Mitch.
And the bacteria that live inside them. And these bacteria take chemicals from the vent environment, sulfide and oxygen and CO2, and they do what plants do in our terrestrial system, and they turn inorganic carbon into organic carbon that we use for nutrition. So these bacteria are essentially doing what plants do for us, but inside the tube worm. So the tube worm doesn't have to eat. And so these beautiful plumes that you see, they're waving around in the vent environment. They're highly vascularized because they are the organ that's picking up the chemicals that transports it to the symbionts.
And what sort of size do they get up to? Like, and how big can these? Because there seems to be some that are on their own, and then some in groups.
Yeah, we think they might be one of the fastest growing marine invertebrates known, and they can grow up to be about 2 meters. Although it's hard to know what their size is unless you pull them up, because sometimes they're smaller than what their tube suggests, because they create false bottoms sometimes.
What are the kind of intriguing questions that we want to understand about how they make a living in this way?
So for a long time we thought that the symbiont was this sulfide oxidizing specialist, and but in the last 15 years, with the age of omics, meaning you can sequence its DNA and look at its proteomics and what genes they're expressing, we now know that the symbiont is far more metabolically flexible than we once assumed. And so my primary research focus is kind of trying to figure out what this bacteria, under what conditions is this bacteria using its metabolic flexibility.
These were first discovered, was it in the Galapagos vents? How did they get all over the world if they're so stationary on a stalk?
Yeah, so they have a larval stage that that is actually symbiont free, and these larvae we think can travel pretty far distances in the water column, and through a process that's still very unclear to us, probably a chemical signaling, they settle at new vent sites, and then once the larvae settle, they take in a bacterium from the vent site, and it's the same bacteria found in all of the tube worms. So there is a filtering filtering mechanism going on that is actually yet how that's done, we still don't know. There's a lot of aspects of this organism that it's still very, we need more people studying it.
Yeah. So do you think it's a shot in the dark when they disperse the larvae into the...
Yes, I think so. But I do think that when they settle, it's because they detect the plume.
Okay.
How's that lighting for you? We had a question about how we identified this as supposed to explore. Can you get into how, at night you guys search the ocean floor for indications of potentially...
Yeah, I could talk through that. Do you want me to... you just want to get out. So, remotely operated vehicle SuBastian makes up about 50 to 60% of the work that we conduct on this expedition. Now, during the other 40 to 50% of the time, ROV Falco is conducting mapping operations. She's outfitted with a really sophisticated sonar system that allows us to map down to about 8,000 meters. So with that, we can cover 99% of the world's oceans, apart from the deep trenches. Now, as well as being able to map the seabed, this system is also able to detect plumes that come up containing gas from the seabed. And now quite a few of these hydrothermal vents are also putting out small traces of gas. Our multi-beam system really detects these quite well. So as we're going along and we see one of these plumes, we tag a location. Now, also, these vents have been previously explored. So we have so we know where these are. As well as going to known vents, we're also expanding out and finding new ones during this expedition. The maps that we're using were also acquired using a special bit of equipment, an autonomous underwater vehicle, which is a kind of self-thinking submarine. Looks a bit like a torpedo, and this one, Sentry, is a unique one which was built by Woods Hole Oceanographic Institute. It mapped this area in November of last year, and identified a number of lumps and bumps which they then explored with Alvin, a manned submersible. But there were still a dozen or so other features that weren't explored, and we're going back and carrying on the great work that this team did by visiting the other features in this area. So that's one of the methods that we use to identify these areas, using our multi-beam. We can also use another system on the ship, our water sampling system, a CTD, which stands for conductivity, temperature and depth, which are the basic sensors that are on there. And we also strap additional sensors on there that measure the amount of turbidity in the water, the amount of particles. So we can go down at set intervals around areas where we think vents may occur, usually in fractures or at the edge of plates, and then we can sniff them. Basically, we look for the telltale plumes that are coming out of these hydrothermal vents, and then we can box them in, basically like a sniffer dog would, looking for areas where there's higher smell of these vents, and then eventually we'll go in and target where they actually are. I'll now hand you back over to Professor John. Ask him how we find them.
Yeah, it's all about sniffing and finding that, well, it's geology too. I mean, we can use the faults to almost predict where they are.
This is what you wanted to collect right here.
This is pink, isn't it?
I don't know. Let's see.
This is 4, right?
Yeah, both. That's bottom of that.
So this is event 4?
This is event 8, and it goes into slurp 4. It's pink mat.
Okay.
Alright. Want to try to get some more, or are you happy with that?
Let's get a little bit more.
Alright.
Yeah, it looks like there's a pretty good interface between the pink and the orange. I think you could get all that without, yeah, I think you could get that without, yeah, I think you can get that without worrying about contamination or cross populating, yeah. Oh yeah, that's a good one. I mean, I'm really fast. I don't know what we're gonna find in these organisms, but I can't even imagine how much, it's a gold mine metabolically. Yeah, that's totally separate.
Someone's asking what degree you need to do this kind of work, and the answer is almost anything you could imagine, right?
Yeah.
Okay, which part of the work is science?
You meet people who are doing microbial, geochemistry. You know, there's all sorts of different disciplines that have to come together to try and figure out what's happening in these unusual environments.
And it's very interdisciplinary and collaborative. I mean, we have experts on board who are animal biologists. We have experts on board who are geochemists. We have people who are microbiology and bioinformaticians, and people who are engineers, who are geologists, and geology and mapping. Understanding the geology and mapping the area is absolutely integral. If we didn't have good maps and we didn't understand the geology, then we wouldn't be able to guide our sampling and target what we do. So I think that's good if we let it all go up into the slope. Here you can really see these filaments. I mean, these bacteria are enormous. I know most of you think of bacteria as these little microscopic things that you can't see with your naked eye. Well, these are incredibly large organisms. Those are individual filaments that you see. It's suspended in the water. It's just, these are, you can see these with your naked eye.
By filament, you mean?
Like a piece of spaghetti. That's what they look like under the microscope, with little sparkling globs of elemental sulfur.
And what do you think?
Can we shake it out and get the rest of it in there? Because it seems like a lot of it's in the tube.
Yeah.
We might have to move to get the white, and we need, we got to do that quick. Sorry, what was it?
And yeah, so the way they're kind of collaborating is pretty interesting, considering there are all these little, what's their motivation to work together?
Well, because you learn more collectively than as an individual. Everybody brings their expertise and their ability to, put a piece of the puzzle together. I don't think, this kind of science isn't something that can be done individually, studying one aspect. I mean, people do that, but I think you have much more chance of discovery of something, really interesting and fundamentally transformative if you do these, multidisciplinary collaborative projects.
Someone's asking how large the team is, and I think there's something like 45 people on the ship at the moment. 45 people?
So, yeah.
Ranging from people who are keeping all those 45 people fed. There's amazing food with the chefs. People making sure that everything is organized and together, and the
What are we doing now?
The deck crew who make sure that all these
What are we doing?
instruments can get in and out of the water
let's move and
safely.
find some white to sample.
The marine techs who
So back
make
off,
sure that we
and I think
go
we're gonna
to
have
the
to
right
go this
places,
way a little bit.
can interact with all the science properly, and can do everything safely and in a controlled way. Then you've got people who have to steer the ship. Obviously, we're sort of a mixture of, you've got to drive this big ship, and then the ship is tethered to this smaller, remotely operated vehicle, and it's a kind of a dance between the two. The ship has to move, and the ROV has to move, and the ROV team have to.
So I think we have to
pilot
back off
this
and go to
small
port a little bit
robot.
to find the white.
As you can see, it's happening now. You've got Ben and Chris who are driving today, You've got Marine Tech John, who is DJing the video feed. So well for you guys, you can see lots of different information and different shot angles. That's all John Fulmer. And yeah, there's just a whole mixture of people that you really need in order to make things happen. Yeah, it's really cool to see everyone collaborate. You don't always get a lot of sleep when you're at sea. You want to make the most of the time that you have, and you know, having supportive people is just the most important thing.
Yeah, and just, you know, everybody has an important role to play, and knowing your role and you know, doing your job, doing your part, and contributing to the collective experience is it's a great example of team building and team process, because like I said, nobody could really accomplish nearly as much as they do working individually. Working together makes you much more successful. Now in this riftia bush, can we pause here for a second and just turn around and get a good shot of this? This is amazing. Look at the map.
So symmetrical.
It's just beautiful. Could we back out a little bit so that we can have that in the shot without the core tubes?
It looks like they're all the same age.
Yeah, it looks like they settled at the same time, and they've got, you know, this is like a canopy with a secondary structure under it. Wow.
Jesse, what do you think when you see those clumps of tube worms?
I am still amazed about how different they are, these Guaymas Basin tube worms, to the ones was previously studying on the East Pacific Rise, a little further to the south. They're just, you know, these tube worms, the oxygen is a lot lower. And even when, you know, we pull them up and we dissect them, their symbionts look different. So it's very, still have a lot to learn.
What would happen if the riftia tube worms weren't there?
Well, they're habitat structuring, so they provide habitat for a lot of smaller organisms. When you typically in the EPR, they're covered in limpets.
All right, let's see. Let's go over this. I think there's some over here that we can get. Maybe. I don't know how hard it's going to be.
Embedded in these gnats.
I'm going to go around the funnel.
Yep. Right to that.
Good. You make that cut. You make it back. Yeah, you make tickle your plastics.
They do provide food for grazers as well, peripheral species that can come in.
It's fascinating to me, though, how you weren't in here earlier, but there was this. I don't know if I took a picture of it. We were at Ace's Spire, right? Actually, it was Ted's Tower, the little mini Ace's, or Alvin Spire beside.
I think I was watching it in the lab. It's gorgeous.
His feathers,
Yeah.
the minerals versus the tube worms blooms, and it was just like, oh my god, fractals and parallels and craziness, and just how is that possible? That blew me away. Did I not take a picture of it? I did not. That was a shame. But we have many pictures of it in the log, but those mineral feathers were just like, what is that? I don't know what that is. It's just beautiful.
Yeah.
And whatever it is, it's forming in that 200+ degree water.
Wow.
You know, up under the flange in the fluid. Yeah, exactly. It's like, wow.
What are the types of
I can't tell what
environments
color this
these guys
is
like to
until
live
we
in?
get a little bit more to port.
Riftia? Well, Riftia is only
So we're not looking
found
for fill.
in the eastern Pacific. So the furthest south, I think, is the Galapagos. but then the East Pacific Rise is kind of off the coast of Costa Rica, and Guaymas Basin is about as far north as they get. There's other tube worm species that are in other vent-like areas, and also are in the same vent areas that tube worm Riftia occupies. But these are definitely the largest tube worms. They're locally known as the giant tube worm.
This is really amazing. I mean, you can see all these colors are biological organisms inhabiting the surface of this structure. I don't see any here. We're going to have to keep panning around. Yeah, we're in search of some white microbial biofilm that looks a little bit like Jello. We're collecting these biofilms to characterize the microbial population and look for interesting natural products and understand how they are so successful in these habitats. If you were watching along before, you noticed that it's hard to sort of suction these off the rock, so they're sticking to the rock somehow. We want to know how they do that. Somebody asked about giant isopods. This is a real low oxygen area. And I think one of the reasons we're not seeing a lot of big animals is because we're oxygen limited. I think we've seen one isopod in the time we've been out here. Haven't seen many isopods. These organisms, oh, here's a nice little Pagoda tower.
So what structure is this?
This is Falco's fountain.
This is Falco's fountain. So this is on the top on the other side.
Yep.
Okay.
Now, if we move down a little, I think we'll see some white gelatinous stuff.
Yeah.
There's the pagoda structure, diffuse flow. There's a nice flow. We're coming over a chimney. There's a flange. Yep. Roger that. These isopods and paralbinellids are just right in the rock, right under there. Can you pan a little bit to port?
What are the little creatures on the tube worm tail?
You know, I am blanking on their name. Pagoda?
It's just
No.
oh, yes. Look right over in here. Can we go over there? Look at these little rifty under here, Jesse. Underneath this is a relict flange. They're all under there.
Wow.
That's beautiful. God.
Can I nudge up a little
Yeah,
bit?
yeah, absolutely. Go for Brad. Get this out of your way. Can you sit that over there?
Yeah, 6 hours of science and exploration there.
Come on. Yeah, keep coming down, keep going around. Yep. If we don't see it here, we can get it at Big Pagoda, which we need to head towards, or we're not going to have time.
What are we looking for?
The white jello mat. But still, I think it's fine. I mean, I think it's, you know, another project.
Yeah.
To really.
Well, I want to come back here.
Yeah. 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 I mean, this is pretty amazing.
Look at that.
No, I mean, this is nuts. Bananas. You just got these little. Look at this guy. Ding 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 ding.
I think we're no longer live
What?
because the video time just got to 4 hours.
Really?
Yeah, Facebook is.
Yeah, it is like 4 hours.
we haven't been on for 4 hours yet. We didn't go live until 9:15.
They just kicked out and said your Facebook feed today.
Okay. Well, that's a bummer. Oh well, that actually gives me more freedom and flexibility. And I don't have to be stressed out.
Oh, I was gonna show my kids that bio box. I guess we're not gonna be live for the rest of the.
Oh no, we'll be live on the SOI feed.
Oh, okay.
Yeah.
Live feeds going. Yeah, it just said.
I'll ask them. I think they're watching now.
Okay, the YouTube one's still
Whoa, beautiful.
Afraid this drop back going in too high. Just keep going right. Take your time as well, so it's really good.
Can you hand the camera to the bio box 3? I think it's bio box 3.
I don't have the camera seat.
Okay.
Just give us a minute.
Yeah, so just
Okay.
trying to get set up on this one.
Some of these rift I've seen today are just blowing me away with their ability to.
So I'm covering it, and this is one of these ones. Right?
The mirror one.
Yeah, live on Facebook again.
And then you got fucking
Live again? All right. Welcome back, everybody. We're here at Falco's Fountain, a pagoda in the Poseidon's Pagoda Valley here in the Gulf of California in the Guaymas Basin.
You'll see the water, it's like jetting a little bit, so you just.
Yeah, we aren't around to it yet. We're close though. It's on this face over here.
Oh, just keep going. Just keep going.
There, it's coming. Oh, look at
Look
that
at
one.
that net on the tip there.
Oh, God, if only we could sample that. Jesus.
So beautiful.
Oh, salivation. Look at that. That is unbelievably. We got some of that though. We have a slurp full of it.
Oh yeah?
Not from there, but the edges are pretty active. Here we are at the, yeah. So let's see where the water's going. Let's try to fire. This is the biggest flange, right? Or is there one bigger on the? There's one bigger around the other side. I can't. Maybe we're on the edge of it though. Let's keep coming around and take a look. Is it this one? I don't know. It's not. It's the lower. Yeah. It's the one. It's below us.
Yeah.
Yeah. It's the next one because this one has like 4 tiered layers,
That's right, yeah.
and we're down. We're up too high. We're
That
at
was
the
the
very.
one that looked like the most floor.
Don't worry about having to loop around. Just getting some great CTD data while you do it.
Yeah. Are we picking up higher temperatures?
Oh, well, you're 3.1, but as you've been going around, so we'll plot. You can plot.
Yeah.
Look at this clump just hanging there.
I know. I saw that one before. And you know what else is different about this place? The tubes look purple, and that's not an artifact.
Try and get a corner and
The coloration is this is a different place.
Yeah.
Well, let me get this in view and then we get in position.
It's been hard to get some of these.
You want to get in position?
They're braided together. The tubes on the bottom, and glued together, so they're using themselves as substrate.
Wow. That's behaviorally really interesting too, right?
Yeah.
Have to grab them today for
Oh yeah. Oh. Oh, that's right. The day we were gonna get it, we had to pull the dive.
hello from there. Oh. Man, watch the book in your eye actually. Just donuts.
we back on IFLS?
I don't know if IFLS got us, but we're definitely live on.
Okay, right? Look up. Yeah. Okay, not yet, right? Let me get in position.
So this is Niskin. This is event 9.
This is event. We need to make sure we're in the discharge. I don't know. Are we in it?
Oh, once we're using the corner, we'll drop down and we'll nudge the corner,
Okay.
which is on the top corner here, and that will get it.
Mandy, this is event 9. You're about
This
to check?
is event 9. indeed.
9, and it's Niskin.
We don't know. Either it will be number
It's just it doesn't look like a slide.
1, 2, or 3.
Okay. What should I call the sample?
Falco's
Yeah,
found. So are we over the big? Are we sure we're over the
Well,
big
that
edge?
corner is over.
Yeah.
If you look over the camera, well, I've got to use it in the arm.
See it here?
It's right, our corner's right on it, right over that flange.
But is this the big flange?
The one that was made the most,
Okay.
most heat.
Okay. Once we do the Niskin, I want to flip around and check the temperature.
Okay. Let's get
Just let me know which one it is.
There you go. Get some.
can't see. Yeah, that's that one.
Move it.
Which one was it?
Oh, can you look at it again there, page?
I'm not sure which one it is. The fire. Let's try to keep position.
It's 3.
3.
Someone's asked, how do we preserve the samples from the bottom considering the pressure?
So if we use this corner as a CTD.
What was that again?
How do we preserve the samples considering the pressure and temperature differences?
Well, the samples are. These are not depths that engender piezophily, so pressure requiring organisms. Most of these organisms do fine at atmospheric pressure, at least for short periods of time. The animals are really sensitive to temperature, so we keep them in insulated boxes called bioboxes. The sediment and microbial samples we get up fast enough that they don't heat tremendously. And remember, they are living around a hydrothermal vent, so they're not just, they're exposed to elevated temperatures to some degree. So we have to, we're very rigorous about working up samples as rapidly as we can to minimize any artifacts like that. Good question.
Just let us know as soon as you do. Once we get this temps reading.
So you're taking the temperature where?
We're using the CDD, so we're going to use the shortest CDDs up here in the vehicle. So I'm going to nudge it in, try and pull the water. No. It's like here. He's so here. You need the basically that. It needs to be looking at that. And that should. We'll see what we're doing. It's
So
really the tight location.
I'll event log the temp.
Yep. That would be number 10.
So I have the lenses on for that.
Bummer. Yeah.
Phil, is that close enough, Leighton?
No, we need to try to get. We're not seeing anything. We actually want to see what the temperature in the venting.
I don't think you're gonna have to get around there now. It's too tight.
Yeah. Are we? We can't get in the CTD flipping around.
Well, that CD's on that corner.
Okay.
It's up here, right?
Ah,
The frame.
gotcha.
So if there's the arms in front too. So. So you hold that there and just lock it in.
Good gracious.
Changes?
Yeah, just keep going.
So what happened to the another heat probe?
Ground fault this morning. I mean, that's like,
Yeah, I'm gonna get up. Change it. No. No, it's not. It's not changing.
okay. So. All right, let's go to Big Pagoda. And I'm just going to run through what we're going to do the rest of the time. We're going to go to Big Pagoda. We're going to catch some water, slurp some more microbial mat, get some riftia for Elva. We're going to potentially pop up to one of those other pagodas to the north of Big Pagoda. And then we're going to come back and we're going to core at the mat site. So Big Pagoda from here is how far?
I'm trying to find it.
It's up to the north, right up there. Yep. Oh, right there. Oop, ew. Over there. That's Big Pagoda.
So again,
Yes.
okay.
And then we want to check out if we have. I mean, we've got 5 hours potentially. We need to, we have to recover the Osmos core, and we have to actually do a. Yesterday, the cores at the Osmos were shot. We did them, it was the last minute, and they were just totally shot. We got to get decent cores at the Osmo site. We got a core at Dan's lander site, and we've got a core at that microbial mat waypoint 20.
Okay.
So I'm gonna allot 2 hours for all of that. I think that should be enough. It's not a bazillion cores,
Okay.
but that gives us how far from Big Pagoda? I think it's about,
It's very far.
yeah.
About 600 meters. It's the thing.
So that's 40 minutes.
I would say realistically, yes.
Then I think we go to the trash heap, and we look how far to the trash heap.
Trash heap would be, I'd say 400 meters.
Yeah. The other thing we could do, Leighton, is look at that feature to the south of where we are right now. Have we been there? That one?
I'm not entirely sure. I think we did when we came through, and there was some minor chimneys because we came from the south originally, I think.
Yeah, because we did. We came
Yeah.
down along there and out.
Yeah, I
There
don't think.
wasn't much there. That's why.
No, you said there was a mountain.
Yeah, there's like a little mound. That's ORP2, or it's
Hey,
close.
what you could do to so you're not just running over like loads of nothingness. Like you do want to go Big Pagoda, right?
Well, I don't know. I mean, it depends on timing.
I mean,
We don't have a ton of
at
time.
these- At this range, it's just running
These
yourself
are-
at the top.
Right, so we've got the middle
Well, you can do the trash heap and the two north of that, which are pretty sure we have it. I just know that there's two more ones.
So we could essentially go up. We could hit the one before Big Pagoda, hit the trash heap on the way back, and then loop back in what, an hour and a half?
Probably. Might be trying to find right now.
So if we did it in 2 hours, that still gives us enough time to do all the coring we need to do.
Okay, so we head up and run along that range.
Yeah.
Big Pagoda. Okay, good.
Yeah.
All right, man. 180.
We're going to overtake the live rocket launch they had on Friday. So that's a good sign.
We did what?
We're going to overtake the live rocket launch they had on Friday. So
That's good. Yeah.
you beat space, which
Yep.
is only
I didn't catch that last time. What did
They didn't know that because they couldn't get positioned as we were.
Did you get some lunch?
I did. I had like 3 bites and ran back. It's fine. I'm gonna get some juice. We have now a bit of time.
That's ship position. Moved. If you could just
That was
make your
great.
ways off to the west.
Good. It was fun. The trash is. Did the people like
Just 2 seconds.
react to that?
Yeah.
I don't know the camera. The camera's over there
Yes.
too. If you want.
It's a difficult one, so you kind of.
Well, it's everywhere, and I think people need to see it because it's just like,
Yeah.
you know, one of my friends that
It's difficult. Who's donut?
Who's donut?
Happy birthday,
Happy
donut.
birthday, donut.
Thank you.
That's the coolest nickname ever. Donut.
What type of donut is donut? Is he a jelly donut or one of those ones with a hole?
It's sweet though.
Sweet, okay.
All got holes in them there.
What?
Donuts have got holes in them.
No, they don't.
Why they do? You've got Tim Hortons, I can guarantee it.
No.
They've all got holes in them.
What? How do they have jam in them?
I don't know how they
Jelly.
engineer it. It's
Sorry.
very well engineered.
Okay.
Just injects it down the recycle.
Is donut named after the like engineering plastic or after the food?
I'm not entirely sure.
Okay. I don't think we got a good sample there.
I
That's
think
a
it's
bummer.
funny. I'm wondering if my daughter had her glasses on.
Had her what?
Had her glasses on because it took a while to see.
Oh yeah.
bridge, ROV.
Yeah,
maybe
it's
in,
great.
speed,
All
please.
3 of my kids
Can
have
we
the
do
same.
0.6?
Like I'm farsighted in this eye and nearsighted in this eye, and they all 3 have the same astigmatism
Okay, thanks.
and the same. It's crazy. And my husband has like 20/20 vision, and mine didn't go south until I was pregnant. I had perfect vision until I got pregnant, and then it just went south and it never recovered. But what's funny is my kids hate wearing their glasses, and they cannot see squat without their glasses, and they're always just like walking around, and then they can't read. My oldest daughter, they thought she had a learning disability. It turned out she didn't see anything.
I stopped reading for ages because I think I just found it really difficult. Just because I was like, I'm long-sighted. Like, just thinking back on it, it was like it just was physically hard for me to read.
Yeah, yeah.
It's
It
really
hurts
good.
your eyes. Yeah.
You don't really notice it when you're a kid. You don't know what something might not be normal about it.
Yeah, my daughter said I wants to get contact lenses.
Oh, my daughter Sophie does too. She's just like, oh, I don't know.
I'm sure it's a safe topic, but you know, just a week.
But it's really funny. I mean, totally funny. Where? I can't even find the picture of what we just did. Where'd it go? Here it is.
So do people transit by those corals that look like?
We got some.
Oh yeah?
Yeah, it's in the bio box. Actually, you can see some of it's. There's a lot of it in the bio box. Like
Yeah.
a lot. Yeah.
The trees.
Yeah. Well, not the trees, but the actual corals. We're gonna try to go by the trees. They're nearsighted. Pseudobasalmica, that's on here to try and get that too. And I need to make sure that we.
Because now I'm curious whether that was actually a coral.
Yeah.
Or was just
15, 20 minutes?
fooling ourselves.
I'll be back in a moment.
Explore, flyer for garbage. We did that. We did that. We did that. We did that. Look at us. We're going through things so fast.
I looked up corals in Guaymas Basin and there's not much
No.
out there.
No, this is the one that I saw yesterday. Little octocoral.
Yeah, I remember. I was on the dive there. Yeah.
This is a pretty one
It's gorgeous.
with a lobster beside it.
I'm glad we didn't sample that one.
Oh yeah, and I can't tell, but there might be. There was something on here that I thought could have been a coral, but this picture doesn't really do it justice, so I don't know. Look at all the little stars on this coral, on the sponge. See that there's 1, 2, 3. They're on corals all the time. I've never seen them on a sponge before. Weird. Put this in my iPad. Here we are coming up on the line of towers. So we're at the southernmost tower. So is this our baby pagodas?
Yeah, so are you gonna go up this right
Yeah.
here?
So we're at a small structure. There's a ridge of pagoda type structures, and we're gonna sort of explore these features to see what what they're characterized by. Oh yeah, this is a small knoll. Whoa, look at the purple. Gosh.
I'll get that line a little bit better.
Wow. Are those ciliates? Yep. Now look at that. Some of them are purple and some of them are white. What's that about?
Some really like prints.
Is the white still ciliate?
I don't know. Hold on. Oh wait, we gotta get that. Look at that. Hold on. I'll stop for a second. It's a nice color contrast. Let's go up and see if the white is dead.
You want to start it?
Right here, straight ahead. Just hold position for a minute.
Looks dead to me.
I don't think it's dead. I think it's just white. It's white precipitate of some sort. All right, let's continue around.
Checking out these.
We're gonna check out these small ones on the way to Big Pagoda and Little.
Yeah.
Yeah, we're gonna do this arc loop back. See that?
So right here.
Yeah.
Right here. Yeah. Coming up there.
And then back down to the valley of microbial mats.
You got pictures of it. Out. 15 meters out.
Polynoids. That was the name. Polynoids.
Polynoids, the scale worms? Yes, that's correct, I think.
That guy said. There is a placky something that has scales down at the fence. That's what it is.
Somebody asking on the stuff, the species genus
Of what?
of the purple.
Of the purple? They're ciliates, and they're do you know their specific name? It's they're called folliculids. So these are the baby old chimneys that are similar to those in Ace's spire. I wonder if we should collect one of these. Maybe not. There's a trash bag. Seems like every beautiful scene that we see today is accompanied by a trash bag. That's a bummer. These are quite interesting. All right, let's continue along the line. I don't want to spend a lot of time.
Hello there.
Oh, wow. That's a gorgeous one.
Hello.
Is that an octopus with a bucket?
What do you want?
I was just saying there was like an octopus next to a bucket.
Yeah.
Next to a trash bag.
Not. If you're on the way somewhere, don't worry about it.
So is this one an extinct mound then?
I think these are just quite young
You can get
features.
The bucket and the octopus in the same shot.
It's a
With that-
trash bag.
That's a trash bag, okay.
Like that right there?
Yeah, that's
Yeah.
wait.
Wait a minute, I know. Yeah, I'm gonna
Yeah.
try to get it more centered and zoom in a little bit.
Just if you leave it
Yeah.
wide for a sec, just
Yeah.
you can leave it there for a sec. It's just a bucket.
You want a little more dramatic lighting?
Sure, always. And you just pull back slightly. I don't want to stress the guy out, so yeah, dramatic lighting is good.
Yeah, it's up down.
Yeah, thanks.
Oh no, he needs to see the spire.
Yeah, if you
Come down.
you can
Come down.
Yeah.
Yeah, that's out of. Yeah, that light's good right there. You want me to go in on the octopus?
I think just get this nice shot of them both together first. I think that whole scene is like
Surreal.
I'll try to get that in.
yeah, if you can see.
Port side.
What's that?
Something on the port side, so no matter of.
Yeah, that's probably that next little tower. We can go check that out.
More church coming up.
So there's that coral.
Really? Oh yeah.
Do you think it's alive?
I do think some of them, yes. Some of them I think are just sort of tumbleweeds and they're dead, but some of them I think are alive. Are we over that next knoll now? This is it? This is that topographic high?
Pretty much it.
All right, let's keep going.
Roger. Something starboard.
Another updated waypoint, Brent.
Whoa, big fissure.
Oh, I see.
Wow.
Yeah.
Oh yeah. Oh, go ahead. That's interesting.
There's a marker on here that says mall chimney.
Says what?
Mall chimney.
MALL? Or small?
They probably meant to put small,
Yeah.
but
Wow. Stop. Hold. I'll stop. Can you back up a little bit? That's your Christmas card, Jesse. I ripped you a Christmas tree. Really? Serious? That's nuts. Shit.
They're up.
Yeah.
We can collect here.
You want to?
How big of Ripia does other one? What size?
No, she don't
You sure?
Ask about it. She never asked. Just
Clams?
She doesn't want Ripia?
I'm not sure. I'm going to ask, but she never
Why don't we
told
just
me.
get some, just so we have some for her potentially?
I think she said she did her last experiment.
I think it's not too big, but
No, not Monica or me. We're
Yeah.
done.
You're good, right?
Yeah.
Look at the squat lobsters at the bottom of this thing are crazy.
Yeah.
We're gonna sit here for a minute and get some nice shots of this.
I'll try to keep it closer.
Yeah. We don't need to, we want to keep it all in the frame, so I don't think we need to get much closer. Because we can.
I know what's going on. The thumbs.db is good, it's got
Can we move just slightly to port, just a tad bit? So can we light the whole thing up? Look at this guy right here. Why is his plume a different color than all the rest?
Yeah.
I don't think that's a lighting thing either, is it?
Talking about the bright red one?
Yeah.
Yeah. It's hard to tell with this camera because I think sometimes the colors
Yeah.
are a little.
Yeah, we're still far away from it, so we really can't
Yeah.
tell when you put it close.
Moving in. And
Yeah, move in a little bit. Whoa.
Might be the light.
Yeah.
It was just reflecting so much, right?
God, now it's just crazy. I'm gonna zoom in and get a close-up of the Riftia plume. Oh, that one has had better days.
I look at the. There's those coated snails.
Yeah. Hmm.
Yeah, worms.
Some of them are so hairy. Look at that guy.
Yeah, look at that.
He's covered.
Diffused flow in there.
Yeah, I can see that. It's
The crabs are.
Oh, look at that hairy squat lobster.
The squat lobsters are eating off of them.
Look at that squat lobster right there, covered in microbes. All right, have we had our Christmas tree fixed? Do you want any of these?
Do you want?
Yeah, please.
Let's get a grab of these Riftia.
And maybe some crabs too.
Yeah, we can slurp some of the squat lobsters.
Big ones or little ones?
Medium sized, and then just a slurp of the squat lobster.
it's the best position for you. Put the starboard arm close to it.
Yeah, we'll just, if you want to sit down there, a little bit, a
I
little
have
level
no
forward.
concept of how big these are. We put the
We can put the lasers on.
lasers, yeah.
Need lasers off?
Yeah.
Yeah. And this is not for doing experiments. This is just for a collection. So you can just grab them at the roots or grab them at the base and yank them out. And the slurp can just be, as many squat lobsters as you can manage.
There you go, Bob. All right, I'll get the squat lobsters first.
This is 10, yes.
So I think we need to come out at 5:30 there.
We need to be over, we have a bunch of sampling to do and a microbial map field, and the osmos have to be cooled around, and Dan's thing has to be cooled
Around
around.
5.
So
Is
we
that
need to be
5
back
okay?
over there. Pardon?
5's okay?
Jar 5, yep. We need to be back, we need to be out of Pagoda land. Was that an octopus? No, that's Riffy in there.
Is that a dead
We need to
one?
be back at, what is that? Let me look.
Like down into your,
Hold on, we're looking at this dead, whatever this is in here. Right there. Yeah, I think. Yeah. Well, yeah, he's pretty much not happy.
not happy, but let's see what he's feeding on. There's a scale worm.
Crabs eating up there.
Yeah. And where's its tube?
Yeah, it looks like it's been pulled out of the tube. How did that happen? Huh.
I'm surprised more aren't feeding on it. Actually. Maybe they scattered. They seem to be coming back as we're watching. Okay.
Are you gonna slurp first or get the rifty first?
Slurp them.
No, it's a dead right
Okay.
there.
You want me to start now or?
Yeah, go ahead. No, don't wait on me. Slurp away. Slurp those crabs. Get 'em.
Can I bring the camera? In?
Absolutely.
All on.
So what container is this?
Slurp 5.
How many do y'all want?
A bunch. 20. 10. More than a couple, for sure.
Turn off.
That's it.
Was that the big one?
Yeah. Ready? Oh, you know what?
that worm right where? Oh, yes, you got it.
Oh yeah.
Did you want it?
Yeah, get more. Get more. They're like, stop it, you big tube sucker. Oh, suck, 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 suck. They're flying.
We gotta turn it off. Yep.
Yeah, pop there. Yeah, get him, get him, get him. Come on, little lobster. Come to your friend. Yeah, let's get those 3, and I think that's enough. What do you think, Carmen?
It's enough.
Yeah. All right, that's it. Two worms. Just a grab of a handful. How many, Carmen? 5, 6, 7, 5. Grab 5. Just a handful of 5 or 6.
You'd have to pour that off the way.
How big?
You know.
I'm gonna put you in there.
It's big, I don't know. It might be. Let's get the lasers. I saw some smaller ones on the side of this clump, so we might want to go for those.
Yeah, that's a good
Depending on
idea,
how
I
big
think.
these are. Big these are. My God, I am totally.
Yeah, I think you could grab these smaller guys on the edge.
Stand by.
In the small bio box?
Big bio box.
Which one are we at the moment then?
We're at, we're not even at Little Pagoda yet. We're just getting some Riftia for Elva.
Look at all those crabs.
You can collect these. You don't have to do these individually. You can grab them.
I was hoping to bundle one
Yeah.
time. I tried
Yeah.
to grab 3 at one time and it wasn't happening.
Yeah.
smaller than that?
Yeah,
There's some
probably
small,
just-
and we can't spend a lot of time doing this, so maybe grab some of these, a clump of these small guys on the edge over here,
Yeah, if you,
like right in here,
yeah.
Oh, okay, we have to reposition for that.
or even down here, maybe. What about these, Jesse?
I was thinking over on the left.
Right, yeah, these
Yeah.
right here.
Yeah.
That's me too. I think those will come off pretty easily.
We're gonna have to reposition. What you
So you need to get a lot of sediment for us today.
Yeah. A lot of take-home stuff.
What would you like?
We found an amazing mat field. I can't believe we haven't sampled this mat field that we found. It's astonishing. And it's on the way back to the lander. It's got orange, white, yellow, and I don't think it has pink, but it has everything other than pink. Yellow, yeah. So we should be able to get everything we need there in pretty short order. Why is the Riftia green?
Green?
The dead one? Is that just a light?
Yeah, I think it's just.
Yeah.
It wouldn't surprise me if they turned green when they die.
Yeah. Sulfur of some sort.
Yeah. But there's even a higher peak for this Riftia mound.
Whoa. That's nuts. Shit, we gotta hurry. We're spending way too much time doing this. It doesn't matter if we. Okay.
2, 1, 2. Someone asked if the team on here were the team that found the Titanic. No, we were just talking in the context of remotely operated vehicles, that they were first used by science organizations really about the early 80s, roughly 1986 when the Titanic was found. We have had some of the team who were involved in finding the Titanic come on board at one point, but our current team are much too young for that, apart from myself.
Where the flow is going. Where's the most active ledge? There's a big flow right there. But isn't there more flow on the port one if we go around a little bit to port? Let's fly around the top and see how. Are we on the INSF?
No, I have this on Para right now.
Para is good. I like Para. Okay.
So this side seems very active over here.
All right, so the water seems to be moving this direction. Let's position ourselves like maybe over here.
How high up do you want to be?
Well, I want to be able to see the signature, so if we can just turn the CTD into it and then turn around and pull the Niskins. We're going to pull both of them here.
We're taking a temp first.
Yeah, we're taking a temp and then we'll pull a bottle.
And that's gonna be
14. Yeah.
There you go. 1.5.
We got up to 5 the other day here.
Yeah, I got this up from low.
You got 5 on the CTD temperature?
Yeah,
Or-
we did.
That's beautiful.
That's an amazing.
Wow, mamma mia.
One right there.
Oh no.
You want to look over at the CTD, see what it closes at?
I'm like right in it.
For the CTD or for the?
That's 5.
5? Okay, let's 6. Oh, mama. That's it.
So do you want me to just sit right here?
Will the CTD, will the Niskin get that? It's coming this way, right?
Yeah, it's
We're
flowing.
all up in it.
It's flowing right through me.
Yeah.
Right
So
now.
6C.
Is the Niskin on the side of the flow or the opposite side?
It's on this side.
It's on that side, but it's flowing to it.
It's flowing into it.
Yeah, but I don't know if it'll get in it, you know what I mean? Because it's got to go up and in it, right?
Well, we can look at it.
Yeah, it's like that.
How high up or how high off of it are we?
We're on top of it.
Okay.
I'm right in the middle of all that flow. It's just coming right across me. It's in it.
See, it's flowing right towards.
It's flowing right towards this right now. Where we're sitting.
See the shower?
Okay, let it sort of bathe them, and then we'll
Okay.
pull them both.
Very good for that.
Which Niskins are we pulling?
Starting with 4, right? And then going back to trying to get one.
Well, we're gonna try to get both of these if we can.
And there'll be 4 and
Just worried they're not gonna. I'm worried the vehicle is blocking the flow into the Niskins.
You can see it like on
Yeah.
see the shimmer? going
Yeah.
up to it?
Yeah, but the bottom of the bottle is below where the shimmer is, isn't it?
It's just, you see where the black bar with the silver bands are? It's
Yes.
just right there, so I mean, it's going right.
Yeah, but it's going this way, and the bottle is situated like this. It's not. It's not getting in the
Well, I
bottle.
mean, it should stop the current right there.
But how does it get past the vehicle? The vehicle's here, right? It's solid. the current's coming this way, the Niskin is here, the Niskin's in the shadow of the vehicle. We need to turn around the other way if we're going to actually get it in the. Can we flip around? Does that make sense?
It does.
Yeah.
I mean, it should, as soon as it stops getting.
No, we've taken a couple like this, and there's nothing in the bottle when we measure it. There's no gas. When we turn around and we get it in the pointed right and over it, it's loaded. I was scratching my head the other day trying to figure out why one of them was low, and it turned out that when it was collected, it was pointing away. Yeah. Yeah, that's more like it. Yep, exactly. That's exactly what we want. This is 4?
This is number 4.
And it's number 15, right?
Yep.
Okay, can we try 3?
What?
Yeah, whatever is left. Yeah.
I guess it's our last one.
I was just gonna be sliding through.
Right.
So I'm gonna go for it.
This is 3?
Our last one, they already broke it.
3, right?
This would be number 1.
Okay.
It's 3 or 1.
Keep them light.
Have a look up there. I don't know what to tell you.
Yeah, but so Carmen, the one that was number 4 in the very beginning was actually number 2.
Actually,
Number 2
Niskin
and
number 1 was example number 2
and Niskin number 3 is
number 3, but
number 3, and then Niskin number 4 we just collected, and Niskin number 1 we're trying to collect now if we can get it to trip.
So it's gonna be these, the ones we found out, that's there.
Maybe putting numbers on the rope where they literally pull it. Huh?
Yeah, on the porch itself, the monkey's fist closest to us is 1, 2,
Yeah.
3, 4.
Yeah.
And then up there it's split different because of the way the wires have to go. So I don't know where they keep getting the different numbers from. It's always been 1, 2, 3, 4 right here.
Hasn't changed.
That's changed the past year and a half. That issue with it. Try to break it.
Me?
Can't try to break that.
rotate, wrap that thing around you, and it might jam on that wire.
I was gonna use it when I opened that vegetable. Yeah.
If you grab it close to the wall where the wire's exposed, will that pull it?
Yeah, and I have the calibration.
It's like a bicycle wire?
Yeah,
So I'm gonna get back to you with the calibration things.
right here where it's oh, the wire. Yeah. Shit.
I mean, I pulled it in.
Yeah.
Yeah, yeah. Okay. Okay.
Okay.
And we'll have a look back up.
16 did not. The number 1 did not trigger.
Okay.
I don't think.
Are we giving up on number 1, Miska?
So, yeah, it's still not fired. Damn it! I wish there was a way that we could pull the stupid pull
It
cord.
just fired. It just
Oh!
Yep, it just fired. Well,
Crazy! Hooray! Yeah. Thank
Get that
God.
wire, pick up.
No, we still got good heat on the.
So eventually.
All right.
Yeah, so yeah, I did. Eventually.
All right. So let's get some video. Let's collect some white gelatinous mat if we see any. And then, yeah, I'm not sure where the best place. Usually the gelatinous stuff is a little lower temperature, so down on the below where the stuff is cooking.
So I'm sitting right here.
I think that is Vegeatoa. I don't know if that's the gelatinous. I can't see through it, honestly.
You know what I'm talking about?
I remember.
On the 4 channels. Then I come up.
Let's move down a bit and see if we can find some.
Okay, okay. Stuff.
So let me zoom in. That's Matt. That's Vegeatoa. We keep going on a port. If you could. There we go, right there. Ring. Wait, I gotta look. I don't know if that might be Vegeatoa. Yeah, that's pink Vegeatoa. Damn it. We have I want to slurp some of the whites. Ooh, there you go, right there. See, it's under there where it's cooler. I don't think that's the same thing, is it? Yeah, that's pink too. Yeah, that's hot. So this stuff, it looks gelatinous.
Oh, like stuff we see on money parts.
Yeah, yeah.
That, like,
Yeah,
those,
exactly,
like, little
exactly,
mounds.
exactly. There's a cousin of that along here, and I think it's epsilons, but I don't know.
So you think it
And
might be like cooler stuff?
I think it's cooler. I think it's lower temperature than this other stuff. Oh, what's that? Hold on. That might be some right there, but I don't know how we would get it. Maybe.
It's a high-rise building. 50 apartment complex.
Yeah, that's not it either. I saw some on the first tower, but we couldn't get in position to sample it. Continue. Oh, look at that. Look at this. Look at this. Look at this thing. Shit. That's totally new. That wasn't here the other day. Dag. That's new from 3 days ago.
Oh my goodness, you bending?
Holy okay, let's get some good footage of this. This is insane.
Yeah, 'cause this wasn't black.
No, 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 it was not. This is totally new. Friends who are watching us. Wow. No, that was not here.
I thought it was that big.
Alvin Spire. Oh, look at the pyrohotite down there. Wow, that's cool.
Oh, that is so pretty.
It is gorgeous.
It's all over. I mean,
Yeah. Pyrohotite, probably. What is up with this stupid?
Wait, is that from,
Right down there, where it fell off.
Yeah.
is this where we were sampling and it
Nope.
broke off?
Nope. That was Alvin Spire.
Oh, that was Alvin Spire, okay.
Just broke off. When you cut off the other lights, which is
Pyrohotite, probably. Yeah, it's similar to pyrite, except it's magnetic.
Oh yeah, there's a mirrored flange over there.
Can we go back up?
real quick, I'm just getting the light.
Yeah, yeah, I got you.
Side. Get the cameras on somewhere.
That's just insane.
oh, the pyrite is just
Pyrohotite. Pyrohotite. Probably. It's like stars in the sky. That's insane. What?
Yeah, because we couldn't see underneath this before.
Maybe it collapsed.
It definitely did. You can see it all down here.
Yeah.
stuff.
That must have been big. Look how deep it used to be over there. That drop to the right, how deep it used to be? Consider deep. Oh. Is it a shallow? Shallow or upside down hole? What is that? Deeper it is, or is it? How does that work? What do you think of? So you got it upside down. So, as you put something in it, you're going shallower or deeper? Is it upside down? What upside down says it already, so we're going.
Deeper, because it's relative.
Of the absolute depth. Of the temperature probe is.
It's like everything has been dusted with fairy dust. And it's just sparkling. This is spectacular. And the black smoke is just surreal. Where's Tom? Is Tom over there? Viet, is Tom over there? Can you give him a yell for me, please?
No, he's not.
Okay.
Oh, the pyrite shining.
Pyrohotite.
Pyrite.
Okay, I'm gonna bring it back up now. Hold on, look up.
Structure.
So pyro, M.
PYROHHO. Wait a minute. PYRROHOTITE. Pyrohotite. It's fire sulfur or something like that. But it's magnetic, that's the difference. This scene is unfreaking believable.
It's insane, you know, how much fluid would this be holding?
A lot. A lot.
Super hot.
All these rocks are hot, huh?
Oh yeah.
Do they stay hot or will they eventually dissipate?
The half-life of these isotopes is. Oh, there's the white stuff, but we can't collect it because it's not.
That's what I'm talking about.
That's one kind of hot stuff. Whoa! Look at this stuff. This is crazy. I'm gonna zoom a little bit up. Hold on.
Right up there.
Now, if we can hold steady. Yeah.
Yeah, just on the zoom apply on.
I mean, this is.
Wait, I'm gonna zoom in more. Whoa! Whoa! Whoa!
Is that pyrite?
Pyrohotite. I'm gonna record myself and push repeat.
Oh, look at those veggie mats.
So,
Hold that right there, that's perfect.
you said this forms when the precipitation is really fast.
Yeah. Yeah. So it's metastable. And it's magnetic.
You want to have a look at the beehive?
Whoa, mama.
Whoa,
Ah. Do we really have to go back to Manzanillo? Can't we just stay here for a few more days?
No one wants to go to Manzanillo.
You know, that's all filled with water, right? That's not actually
It's not
exposed.
in the usual world.
Yep.
Yeah.
Anyone know where his cabin is?
Where's Kevin's at? He's the one right by the sauna.
Can we do that again a couple times? And we're gonna try to get the mirror effect in this cavity because there's just a ton of water coming out of here, and it looks like it's empty, but it's
It is empty.
well, except under the backside where it's still covered up.
Broke off right
Yeah,
there.
yeah.
So it used to hold in that much. The mirror.
Can we put the lasers on that and get a size it, and then we can figure out how much volume is there?
Just look at the inside
Yeah,
of this
yeah,
and the shine. It's.
Did this just break then?
It's broken in the past couple of days.
Yeah, see the new
That's
beehive
the
and
rock.
everything up there?
Oh,
Pretty, that's
pretty.
pretty
But
heavy.
it's got to be hot, right?
Is this big?
Oh, yeah, we can't collect any of this. It's radioactive as all get out.
Radioactive.
Yeah, totally. Totally.
It looks awesome though.
Crazy.
Gorgeous.
Almost perfect to get all those rings in there now.
But the black smoke is the thing that just makes this just wicked. And this little feature has precipitated in the last couple of days. Yeah, let's get a couple of shots of the beehive.
Lasers on it. and figure
Yeah,
out-
yeah.
Whoa, look at that feature right above.
I know, we're gonna go up there too, because that's interesting as well. I mean, there's just black smoke coming out everywhere.
This is just madness.
Well, we've now documented it. We've got before and after, so we can compare.
The heat signature around it's much bigger.
Yeah.
Yeah, we're at 4 degrees this far away.
So the beehive is about 10 centimeters.
Kind of a bummer we didn't pull the Niskins here, huh?
Well, I think all this hot water was coming up there.
You think so?
Yeah, I mean,
All right.
that's like see right there.
Whoa, look at that chimney.
Because it was significantly hotter up there than it was the other day.
Yeah.
We were reading 7 this time instead of 5.
Wait. Solid. This is where Millennium Falcon was.
Oh, so it's all Millennium Falcon.
Did Millennium Falcon shear?
Yeah, that's where this.
Oh,
Let's go see if-
bummer. Let's see if he can find the crash site. That's a.
Is that part of it to do? Everything has little gold shivers on it.
Yeah, it's all been dusted.
See, look at all that down here. Yeah.
Look at all the fairy dust. Wow.
Oh yeah, pieces everywhere, yeah.
There's that piece right there. It's all pieces.
You know what it looks like when somebody breaks into your car right outside?
Oh, look at this spire. Yep, that's part of the Millennium Falcon right there.
Wow. Yeah, it just
There's a new little
blasted.
That flange is called
Millennial Falcon. Yeah. You know, it did look like it, kind of.
Yeah.
It did.
That's new.
This is new?
That orifice, yeah.
10
wow.
centimeters. 50. I think that one that's unfinished. I don't think it's
Yeah, that's definitely
crazy.
Millennium Falcon.
It is. I think you're right. What a bummer.
Because we'll come from it this way. Wrap around. Because there's your runway.
Oh yeah, you're absolutely right.
That's the runway right there.
So we can totally do a before and after and compare the features.
Hey, 'cause
Yeah.
Look how. You remember how clear it was here before?
Yep, and it's totally turbid now. We really do need to get some fantastic, just digital, high def coverage of that whole thing so we can do that comparison.
That.
I like the way that's curving as well.
I mean, it's.
mine.
Yeah,
What? It looks like
Give it
Florida.
better runners than the website.
That one blows my mind that it's stable out there.
Yeah.
It's just like,
It's hilarious. It looks like
the hot
Florida.
fluid has to contribute to it. It has to. I did some calculations. It's not impossible. It takes a lot of volume, but looking at the cavity there, it's huge.
It's huge.
Yeah.
Yep, that was definitely it.
Wow. Did we sample on Millennium?
We
Falcon?
did sample, but we're done with bottles now.
But we sampled biomass? Like the,
We didn't collect stuff from the top of it, or the bottom,
so
actually.
you don't think we contributed to it falling down?
I don't. I think this is just part of the natural process of how
Yeah,
this
if we were
system's
down
work.
there when that thing collapsed, we would have known
We're probably.
maybe lost
Yeah,
some senses.
but maybe we pulled the Jenga piece and then left before the big event happened.
When was the last time we were here? 2 days ago?
3 days. Wednesday or Thursday.
Is there no way we can get any part, right?
We have some of this stuff outside. It's
But it-
really hot.
Oh,
Yeah.
like I'll have my future kids coming out at 6.
Yeah, you don't want them having. Yeah, you don't want that. This is just insane. Yeah. Insane.
I wonder if it destroyed our little ledge we put the rings on.
Because if they do,
Oh shit.
Let's go see.
Yeah.
this is the only
That'd be
explanation.
where,
my wife's gonna point out this and kill me.
But that's on top of, it all came down on top of our little,
Yeah, but we did the rings. Nobody else took the rings there.
That's insane. That's, that,
well, that's Millennium Falcon. I mean,
there's more that fell down too. Because remember, Millennium Falcon didn't have Riftio on it.
no. It probably took everything down underneath it.
They were ripping down.
Yeah, they were. That's insane. That's still all Millennium Falcon.
Look what I did. Look at this slow-mo. It makes it, it's the,
Oh wow.
You're talking about the dust.
Cool.
Some of the rocks.
Not these.
Not these, no, not
yeah,
these.
because it,
But like the sparkles.
These are-
Because of the light
Yeah,
variability, it made a strobe effect.
That wasn't there.
It wasn't even on purpose.
This is where we went in to put the rings in.
What is it, just above it? You're looking at this area right up here.
Look, there's a runway.
That's up in here. That's right there. Right where all the black.
Wait a second. Oh, there it is.
okay.
That's
A ring pool has been protected.
Okay, it's on the other side. Gotcha.
there is dust on it.
Aw,
See all the black dust?
see little baby mirror flanges.
That's where we get the rings.
That's where we forge the rings.
Oh yeah?
Yep.
Oh, that's so pretty. A baby mirror flange. Shit. That is.
Might still be light.
It is live, but it's okay.
I've dropped a couple myself today.
So,
Oh, I didn't
Oh.
drop anything. I just that was amazing. This time I was a little
okay.
well. What the heck behind like that? I've realized this morning.
Yes.
Alright, continue looking for the piece just fell off. Piece just fell off.
Whoa.
Nowhere near it.
So let's what? It's got to be just
It's trying to
so. I'd love to have like a camera. Look at the. Let's just sit here for a minute because somebody can do PIV with this if we get good footage of it and tell us what the discharge rate is. Let's let it clear up for a second. This is worth getting some good shots of, and then we're going to be off to hunt for new pagodas on the pagoda path.
We have to, when are we gonna collect the sediment?
We're going over there now. We're headed by there.
Oh, that is,
What?
That is amazing.
Stop. Can you back off just a little so we can get a wider view?
Hey, can you look to the left a little bit? So we get the lights in it up?
Yeah.
I'll get it back. More to your left. Right there.
So what's the brown color of the? Is it just iron sulfide you think, or is it something else precipitate?
Hard to say. Iron, manganese.
You think any iron oxide?
Oh yeah. But it has to be mixing with seawater pretty rapidly. But I mean, it is. If you look at the, I'm gonna send this to Tim Crone and to a couple of other people because I think you can. This is insane. So we're at Big Pagoda. There was a flange collapse here in the last 3 days since the last time we were here. We used to call this the Millennium Falcon flange, and the Millennium Falcon crashed. And now we have smaller flange with rapidly accreting mineral towers on the top of it. It's just phenomenal to see. And black smoke coming out of the top. It's got to be super hot fluid too, right?
So do you think the flanges fall off? And
Oh
then
yeah,
get
yeah,
remade
yeah.
as
I think,
the fluid
yeah,
leaves again?
yeah. I do.
I wonder,
It's an upside down waterfall. Yeah.
is there any way to test, like if you sampled the edge of a flange, how old the precipitate
Yeah.
is?
Yeah. Strontium isotopes, probably. Strontium. And you could use, you know, radium and uranium series too to get an idea, and C14 obviously, because it's not,
fast waterfall.
you know.
That's so gorgeous.
Wow. Yeah. Can we just sit right here?
That looks really cool.
Wow. God. Shit.
It really does.
Brother.
Look at it upside down.
I'm going to. Right now, I'm just too mystified by it all to. Can we back off just a twinge tidbit? God. I mean, for all those people not watching, I feel sad for you to not see this for the first time along with us. God, this is crazy. Okay, can we go back one more time? Kind of down and then up. Okay, hold on there. This is just spectacular. Boy, oh boy.
You can't put this in words.
No, you really can't. This is just, their words don't do it justice. It's, whoa, stay right there. Holy cow, hold that. All right. As much as I would like to stay here for a long time, we have to start making our way back. And
It's just feet gorgeous.
yeah, yeah. So let's head. Jeez. God, that's just crazy.
Yeah, this is one that we looked at. We're like, oh, I wonder how deep that thing goes.
Yeah, well, now we know. Now we know.
Now we know. I know. It was about a meter deep. All right. Where are we going now?
Can you back off the map and I'll show you. We're going to the little unknown, undocumented pagoda between to the one right beside. Yeah.
These two guys right here?
Yeah. Well, what about the one? We've been to Trash Heap, but not to the one beside it.
So I got like three right
Let's
there.
try the one right there, and then we're gonna go to Trash Heap, and then we got to go back to the mat land.
You're going directly east. Geology.
I see your updated waypoints.
Thank you.
Thank you.
Good. Moved off right now. Go.
Stop, wait, stop. Damn it. This doesn't get like 1,000 or 5,000 retweets. There's no God. We have trash, right? We've collected trash. Be right back. Thank you.
What is that thing? Is that from? That's a wiki page.
Yeah, I'm looking at.
Huh? Is it? Yeah, someone running it. Seen all the time last week. I feel it.
Oh, look at this beauty.
Looks like they had Osmos.
Since why are they about that far?
I believe, yeah, that would make sense because
Is the lander over here?
Oh, it's way down there. Yeah, we gotta go back over there. That's where we're going next.
it's pretty close to Dan's tip.
It is. It's all close. We have to stop by waypoint 20 on the way back and map core. We'll be all right. We got 3 hours. That's why I'm tracking the time today.
Yeah, we got distracted by beauty.
That was insane, though. Good Lord.
Scientific beauty is fine.
You know, the funny thing is that what we're gonna find, the last thing, is a big packet of mussels. Like all the mussels in Guaymas are gonna like appear. Whoa! Hello, Pagoda. Maybe this one should be Rifty as Pagoda. Jeez Louise.
Yeah, look at that.
Good.
This like
Okay, people online, now's your chance to name a pagoda.
rent-controlled Rifty apartments.
Look at the back. Can we do a once around of it and get some video?
This is little.
No, this is unnamed. Nobody's seen this one before. This is a first look. We had some good names the other day that were suggested. This is the new one.
Oh,
Huge
sorry,
amount.
buddy. Just so you have.
Huge amounts.
So this isn't garbage, right? This is the one.
No. This is the new one. Yeah.
Do you want to put it out to the audience?
I just did. I said, whoa. Not yet.
They asked if we've ever gotten seasick.
There's a
Oh, there's a
marker.
marker.
Somebody's named it before us.
That's an old marker. I have no idea. Can't read it. It looks like
It looks like a marker. I mean, it's
there's an octopus.
Plenty of winning on it.
Yeah. There's an
It's almost the science sample itself.
Is that a trash again? We may do some more trash collecting. There's I may want to collect another one of these trash bags for.
where's the octopus?
I thought it was an octopus. It's a trash bag. It was the right color.
Some of your naming ideas. Turtle shell pagoda, tectonic pagoda,
They need to have the first letter in the first and second part of their name, like Falco's fountain and Rebecca's roost. This is like that game, the Puzzle Master.
Like Pagoda Palace.
Yeah, Pagoda Palace, but except this is only one. Riftia Ridge. I like it. Riftia Ridge.
Riftia Rump. It's not a ridge, is it?
Okay then.
Rifty a rabbit hole.
Rifty
Something
riot.
dumpling.
Rifty a rise.
Riftia Rise.
Yeah.
That's a good one. Going once. Going twice. Riftia Rise. I like it.
Hey, is that fried egg?
That is fried egg. Yeah.
That's fried egg.
It's a fried egg face, actually. It's like fried egg cat face. Look at it. It's like a cat. Upside down, but a cat.
Bacterial. I like bacterial. That would be another good one.
Sebastian slope.
Oh, Sebastian Slope's not bad, but I like Rift Your Eyes.
Yeah, Rift Your Eyes is good.
What do you think?
Unless anything else goes. Tectonic Tumbler.
Turtle top.
Turtle Top.
Tectonic tumbler.
SuBastian slow.
Oh, look at it cracked. Turtle is a good one. Turtle Top Pagoda. I still like Rift Your Eyes. Wrong with that app? Motion sickness would be a big issue then. Yeah. It looked like a snow globe driving around in 3D,
Yeah, we're gonna have to start booking. We're gonna be able to spend like 5 minutes at these other two each. Oh, hello, Mr. Octopus. We cannot stop, and we're gonna have to just get him in passing and give him a flyby hello. Hello. Goodbye.
Probably friendlier anyway.
It's probably gonna make him very happy
He's
tonight.
been pretty friendly. Pose for the camera. Even sit inside a balloon for us.
I worry about their eyes.
once saw me and all.
Check them out.
I do too, because you know that can be
Octal mount modesty.
89888.
Need to find one with loads of rift you on and then call it Joy's Jungle.
Oh, that's a good one. I like that.
Joy's Jungle.
Yeah. We'll do the next one.
Or Jungle of Joy.
Yeah.
The Jungle of Joy is a good one.
Oh, that's even better. You are so clever.
Oh, we've had plenty of time.
Yeah. Keep 4.
Where y'all going back to that,
Who is Rebecca?
Yeah, when we went.
Rebecca's roots.
I don't know. That was named long ago, and I'm not sure where it came from.
He would.
Probably someone's kid.
Yeah.
Oh, yeah.
Yeah, he went there with
We'll be honest.
You with them?
yeah,
Oh, yeah.
John.
Yeah, it was
But
good.
he doesn't like seafood. He loved it. He was bringing him, like, I guess, his buddy and gave him a look of juice. And put him in.
I was thinking we'd have to give Ben like CPR because he had some spicy stuff he looked like was on his way out.
That was hilarious. That's just been. It was that one show you people.
Wow. This is a
Okay, we get to name this one. This is a new mound, a new pagoda in the Valley of Pagodas.
It has some flanges.
It has mirror flanges, it has riftia, it has diffuse flow.
You have to go with the character of the vent. It's got to.
Yeah. What is it telling you? Can we go around and see the other side and the top of it?
It looks like a mushroom.
Magic mushroom vat. Nah, that doesn't work.
This is the one
That's,
slightly.
wait, you know what? There's also something back here. Look at that thing. It's like a volcano sitting atop it. What? Oh no. Okay, let's get some video of that one. Jesus. I mean, these are really Jenga structures.
Joy's Jacuzzi Jungle.
Joy's Jenga jungle.
Jungle of Jacuzzi Joy.
Joy's Jacuzzi.
Joy's Jacuzzi.
Oh, Joy's Jacuzzi. I like that. Wow. Look at the one to the port. This is massive. Oh, look at. Jesus, that's amazing.
Floating flange.
Oh, wow.
No, it just came up as like floating flange.
Wow.
That is epic.
Criminy.
That is
Highlights coming on.
Yes. Oh, highlight. Jeez.
He does like Joy's Jacuzzi.
I like Joy's
Joy's
Jacuzzi.
Jacuzzi's a good one.
Joy's Jets are also good, but I like Joy's Jacuzzi better.
That is epic.
It is.
Okay, I'm happy with Joy's Jacuzzi. Is everyone else?
I think so.
Joy's
do
Jacuzzi.
it.
Yeah, look how good that mirror is right there.
Wow.
That's what he's making one of.
There you go. You need a t-shirt now. I went on this expedition and all kind of came back with it.
It was a geologic structure named after me. Look at this guy. This is pretty epic. Wow. Wow, wow, and wow. So this is 21-23.
Well, we're all gonna get to say we were here.
Yeah. God, this is.
It must have been like that.
What?
That is epic.
That is totally epic. We got to do one more up and down because this is just.
Clear shot of inside. You see it's still working with.
Yeah.
There's so much we haven't seen yet.
No. There's so much we need to see when we come back.
When we come back?
Soon, I hope. Very soon.
Few proposals, right?
Yeah.
Write down names for different pagodas.
That was pretty cool.
That's very cool. 27. Next.
There's all over the top. See what the temperature is. 3.3. I like Joy's jacuzzi.
I like Joy's jacuzzi either. I like that too.
Alright, you ready to throw to the next one?
Yep, trash heap.
So much we'll get there now.
Looks like I got one little mound to go in front of me.
Yeah. Oh yeah, we weren't even on the big mound. Good grief.
Wow.
Yeah. Oh, this is rubbish. Right.
Is this trash heap the backside?
Upside down one? Four. Yeah. Squashing is you inverted. Is that just a ball of 50?
Yeah. Surrounded by Matt.
This is the back side of the trash heap.
There's a bunch of ribs coming up.
That should be to your right.
Whoa.
That's trashy.
Whoa, look it. That's some riftia.
I'm gonna film that.
Highlight for sure.
This is baby Pagoda? Where are we?
This was trashy.
This was
This is trash heap, yeah.
And it says there's a garbage bin, which is a trash heap.
Trash heap is beautiful.
Yeah, but at the base of it, it has tons of trash.
It's like ivy climbing up a house.
Yeah.
You just took some of the yolk off the fried egg.
I mean, the riftia is just like a coat, girdling the whole thing. It's beautiful. Look at all the babies.
I know.
That sounded like I know, I wish we had more time.
Yes.
Yeah, like another 2 weeks. Like another, I want my 2 weeks back.
Oh, which one's that?
See, there's right in the middle. There's a little crust.
What? Whoa, that's different.
That's when I was just coming up.
Yeah.
So, is that like a little pagoda to our trash heap?
Look at all the riftia here.
Oh my goodness.
Wow. But the temperature would not be a good one when we went over. About 3 heats dropping out.
Can't wait till you plot these temperatures up. Whoa, mama.
Okay. I will use it then.
You can use it. Just plug it in.
Screw the crouch. Which one are you looking at?
Second from the one on the right.
Oh, whoa.
Yeah.
Oh, hello. Objects seem to be a bit may appear close to them.
What is that in the distance behind there?
Where he just was at.
Okay. All right, so maybe which? So we're not even to trash heap yet. This is the.
This could be like a mushroom field, right? With all these mushrooms. Fairy ring. Is the
Okay, mushroom field.
mushrooms. Fairy rings. Don't never use that term.
But we also don't have a unicorn as our mascot.
What's wrong with that?
All All right. I think we take one flyby trash heap to get the trash, and then we got to run over to the other.
That's trash heap, right?
Yeah, at the base. Yeah.
So I think we came from the opposite side last time.
Right. How far up does trash heap go? Whoa. Trash heap goes up a ways. We still on paradepth? So 15 meters, that's pretty good.
Jarby.
Thanks.
It's a big old lump, really, isn't it? When you look at it.
Yeah.
It would be amazing to think what that looked like if you took it out of water.
Mm-hmm. That's a lot of riftia. Good gracious.
Dead on the, we've heard most of that on the bottom.
So on the side, right leg, on the track.
Wow, look at that.
Look at that flange.
Oh, you can see where.
Whoa.
Now that is
Whoa, that's crazy.
that would look good as a potted table. I mean,
Like a live edge table.
yeah, with just
Oh, that
potted.
would be such a good live edge table.
Oh, yeah.
The radiation come out of it.
Yeah.
That is cool. You're looking into the cauldron of life. Exactly. Yeah.
It's pretty hard, right? I mean, that rock is.
Hard, yeah. I think it's pretty big around.
Can't actually see any trash.
Yeah, it's not something like that.
There's some of it.
Just more purple stuff like that.
I think it's down. It's on that. It was where we approached it from over here. It's on that line over there.
So the actual tubes of the tube work's really tough then, because they seem to litter the place.
Chitin, right?
Yeah.
I'll spill a lot of rips in that.
Look at those sponges.
What are those?
Anemones, those are sponges.
The white blobs?
Yeah, I wonder if we should grab a couple of those. Look at this riftia again. Good lord. We should not grab the sponges. We gotta go get mud. We're gonna run out of time.
Yeah. Yeah.
See if we were naming this again, because we haven't seen any rubbish. Rampant Biftia would be the one.
What?
Rampant Biftia.
Rampant riftia. Yeah.
Oh, like pasta on rift.
Hasta la Biftia.
Fighting fight. I like that. Fighting fight.
It's quite a
Yeah,
tall
I
thing.
think she's right. When we came in, we came from.
Okay, this is a different
Yeah.
angle there.
Yeah.
And it was all the way at the base. Like a little tumbleweed stuff going on.
Ah, yeah, that's right.
Might be to your left.
But we go all in mud.
This thing was pretty big.
Yeah, it really is.
Did you want to go look at the trash or?
We kind of need to boogie unless we go right over it. We need to be over there by 4.
I got the vessel moving right now, so you can bring up
Okay.
Does anyone need anything?
Nope.
It's on that shadow. It's like, there's a shadow coming in there. There's another one.
You came all the way around. That's the other one we were at.
No way. Well, looked at pictures last time, but never mind.
Yeah, to the southwest.
Oh, Ben, Ben, do you think you can sit down here for like 5 seconds? Things are on. I can't have my glasses.
Is that not?
We're jelly, yeah.
I don't have anything on.
Oh, thank you, sir.
Okay. I do not understand why.
Oh, little white chip. I don't like.
Huh.
Oh. That's spider on that one. It's pretty impressive.
For a lot of people, this is reality TV.
Yeah.
I love it.
Oh, look at this.
Yeah.
How tall do you think that is?
Yeah, not very.
The Empire State of the Deep.
It's like more like the Burj Khalifa in Dubai.
It's Canada.
Yeah, Burj Khalifa is probably a better anecdote. Yeah, I would just keep on with all that.
Also, what is the depth, direction, and heads up related to this heads up display stuff? It's exactly what you choose.
It's all gone.
Oh, well, it's like really, yeah, I'll be like,
Have you seen the Burj Khalifa?
I haven't been in, yeah. I've seen it from the air.
Massive. Will you update his waypoint? We're going down over here. Pretty much following those lines.
Heading southeast. I'm gonna bring him down here. Great, do you have a new waypoint?
thanks.
Anybody wants anything in the galley? Who's going down the galley? Andy?
I'm good, thank you.
Me.
Yeah, I'm looking forward to that restaurant.
Yeah.
That was good. So we gotta do restaurant,
Ice cream.
tequila. Did you need to find tequila? Oh, like the big flops. Yeah. You have no issues with taking that.
No, it's not good.
Oh, okay.
Tacos. This is the taco stand behind.
Yeah, street food.
What do you get? 6 tacos and then food's right.
Ciao. Well, I took out everybody's day.
Taco coma.
I just want to sit in the sun. I feel like I've been like slightly cold.
I've been freezing for like days now.
There was a sauna on the ship.
Just can you compare?
I bet the sauna gets a lot of use when like the ship goes to the Antarctica. Such
Yeah.
places,
True.
cold the North Sea.
I mean, we just haven't had any time to go to You know, the amount of jelly here like really reminds me of that place in Palau. It's almost like a jellyfish lake. Number of
They all
jelly.
seem to be the same species too.
Yeah, they're
So I
everywhere.
don't know if there's like a bloom that happened.
Could be an oxygen thing. Could be a bloom.
Good point.
So, what are the predators? Who eats you?
Down here?
Turtles still?
Down here, nothing.
Oh, nothing. Yes.
Yeah.
Explains why they're so bad.
What about octopus? What are they gonna?
Fish, crabs.
Yeah, but I
octopus
mean-
is
Yeah.
But I mean, down here, there's not a whole lot but the squat lobster.
Yeah.
Oh, we did have a octopus and a squat lobster on video going after each other. And the
Yeah,
octopus going
yeah.
after the squat lobster.
That was a good
Octopus don't go well. They do go after fish and sharks
Yeah.
as well. So.
So we're gonna head to. I think it's waypoint 20.
That's where I'm at, right
Yeah.
now. Right over by the lane, right?
Yeah, but it's a big mat area. It's a little bit away from the lander. I think it got moved. It's taking forever to upload. It's been uploading for like 10 minutes.
As soon as we finish this, we're gonna box it.
So slow.
Tweets?
Yeah, I put
Yeah,
it.
it's been the videos have been taking off and off.
I'm glad we went over there. That was a trip.
The other thing was cool about plotting the tracks is it tells you how far the ROV actually travelled
Mm-hmm.
during each dive. And we covered some miles actually. When you go in Alvin, it's just a real careful balance of batteries and how far you want to travel.
Yeah. If
Yeah.
you drive, it's
Guzzles the batteries.
guzzles the batteries.
You can't use the manipulators too much.
Yeah.
because They're run on hydraulic.
Yeah.
So the batteries power the hydraulics. Oh,
Yeah. Yeah.
Like electric arms.
What about the ROV? Is that just powered by the tether?
Yes.
Same,
HVU on that. 4,500 volts.
the same tether that has the fiber optic? That's crazy. I feel like that it's not thick enough to carry all that.
5 conductors, 3 drain wires, and 6 fibers.
No. How often does the fiber optic break?
Shh.
Not very often.
Not very often. Not on this style. This style of umbilical, since it's armored, it's really hard to bend
Yeah.
it. But some of the other ROVs use a soft tether and the TMS. Those fibers break.
Yeah.
And all 6 of those fibers are in a stainless steel tubing.
Yeah.
That are the size of that yellow fiber that's poking out.
Yeah.
So they all get shoved into a stainless steel tubing. So but if a conductor does blow, it will shatter all the fibers.
Which we had that.
Because it's all twisted together.
Yeah.
And then the fibers are in the middle, the stainless steel tubing. So
Yeah.
if one of the high voltage
mines blow, make a huge explosion inside, and just shatter it all.
And then you look at something like Nereus, which was, so it had a steel armored umbilical down to 6,000 meters, and then a canister, like a depressor weight, but that would split, and then it would fly around on a hair thin fiber optic cable. So the vehicle had the batteries and all the commands like this went through that cable.
Yeah.
It was designed for like torpedo tubes. You can imagine how often that broke.
Who is Nereus?
Who was Nereus?
Who had Nereus? Yeah.
Who we had Nereus. So we used it for a few cruises.
That's
we took
that's
it
the
out.
tube that the fibers come out of. So that's all the
Anyway,
fibers right there.
probably.
So they come out of a metal tube,
Yeah.
and then up here these are our conductors that are all in it.
That's cool.
I'll show you when we get on that.
Cool.
We used to have some sections. You came up with a big bag full of them.
What's that?
The umbilical.
Oh, when we had to cut off.
God, that's crazy shot.
Miles. 10,000 big 80 million.
What did they think when you took that to the airport? Did they say anything?
Oh, yeah. Oh, Chuck, I got a question. That was the only thing that was in the back. Was that and some fuel pump?
And this group of sites is going
Yeah,
to
like, look at this cable board.
be in scope for like the next decade
Yeah.
as far as Guaymas
Yeah,
Basin go.
I think so. I think so.
How would the traditional Guaymas sites compare to these?
Yeah. We've lost 5 valuable minutes messing around in my stupid spot. I think all this may be rocky. I mean, we're not at the spot we saw. I mean, this is close, but it's not. The spot we saw was just flat. And no venting, obvious chimneys, little diffuse flow, but not. It's a bummer.
Anywhere over there, just to see?
Yeah.
I want to go on the very far edge just in case it does go down.
Okay. Oh yeah, sweet. Business. Just leave it in there. We'll get another one.
Okay. I'm gonna go a little deeper because it's really soft.
Rosa, can you give me one imaging from this screen?
Sure. Which one is? Which one?
I need one imaging from
Oh, an image?
this.
No,
Oh,
no.
yeah, please.
You got a phone?
Yeah.
You take a picture of it?
I need this for my report.
I know you can yes, anyway.
So you just want me to periodically prove anywhere?
So if you sort of start over here and just sort of work your way down and across, you can do it with.
along the ridge or just?
It's a little bit from the inside. Like right there is perfect.
So, is this still at Mount?
Yes. It's a different location, but it's a.
We're just on the other side. See that chimney right there? We were just on the other side of that. So.
Okay. And this is event
Yeah.
7, the first core is event 17?
Whoa! Excellent. Excellent. This is awesome. Trillium. We back off the light just a little. The oil seeping out of that.
Number 16.
Yeah.
Number 16 is epic gateway on
And then you
want to get an image of this, right?
Yeah, just so I can make a core map.
You want it further out?
Yeah, yeah.
I can another shells for you. This one here?
Or do you want this one?
That's good. We don't need them super deep. So that's 8.
It's really soft right
Yeah.
there.
So yeah,
That was 12 feet.
12 feet.
and the first one was 10, right?
First one was 15, then 16, then 12 feet.
And Mandy, you're starting the cores at event number 17, right?
That's correct.
1416 copy.
This is gonna be oily too.
Oh yeah, they're all gonna be oily. This is fantastic.
That was 14.
Okay.
Thank you, Ross.
That's you too.
Number 11.
Good. Oh, look at the oil. going. We are collecting sediment cores in a very oily, probably hot microbial mat on the edge of a fumarole field. These mats will have a lot to tell us about anaerobic hydrocarbon degradation and potentially reveal some novel organisms that carry out this process. After this, we're going to go collect some instrumentation and collect some additional cores over next to the instruments. Except today it's number 8 instead of number 4. It's the quiver that's, they're not attaching the quiver in there properly is the issue.
On 36. Or not.
$36.
And then you want this one a little closer to this, correct?
Yep. Or as close as you can get to the other one is fine.
Okay.
Yeah.
Just on the white.
Yeah.
Just 4, correct?
4 total, that's it. Yep.
And I'm 36.
The what?
The thing I just apparently I can't hear a number being called.
Is that something stuck in the thruster?
Plastic bag.
That's right. One of the charge things. Well, that's just that fish.
I don't think so.
So, where is Mr. T at?
Mr. Who?
Dan's sampler.
I know I've seen it.
Is it over where it's labeled Osmo sampler?
I believe so.
Okay.
And the lander is right behind it.
This one does not want a seat on its bung. Come on. Shit, it still hasn't gone down. I didn't see it. Did you see it pop? I think it went, it's still loose. Damn it. Yeah, try it down there. If it's not moving, it's not gonna move. So how do we, is Dan's thing just gonna be bungee down?
Mm-hmm.
Okay. You're in. Oh, a little bit down.
Yeah, all he says is take more end of time series screen grabs. Yeah, that's it.
Okay. He does what cores though, right? I'm gonna get cores
I'm
because
assuming.
otherwise there's nothing to pair with it.
Yeah. He did not specifically mention cores.
You know, we didn't do that in the beginning because he didn't mention it, but now that it's changed so much, I'm kicking myself because
Yeah.
but we do have cores from nearby in maps, so we can kind of sort of maybe say something.
I mean, he's still gonna get a picture of the micro habitat
Yeah.
profile.
Yeah, but it'd be nice to have some chemistry with it to
Yeah.
compare. We can do the chemistry, it's no problem. Either way.
So good to pick them up, right?
Yes, close the valves first.
Show the very good one. Should be. But firebox. We're just gonna get us a little bit closer. Yeah. He said there was a way to
You want
get him
these
to
logged
show
as
me
events, right?
The recovery?
Yeah.
I did try to do it. I could sell one. What's it? Putting my master control book. With pink bands, yellow and blue.
How's that? You want me to get closer? You guys have to manage that one. Remember, we're gonna close that valve there.
Oh, a little bit more. It's got to stay over there.
You jam on to the end.
That's it.
What's that?
See that?
beginning to like tap the handy, paste your pictures right there, change color.
Yeah, it's really, I love it. I mean, it took me a while to figure out how to really utilize it properly and get the most out of it, but boy.
Copy and paste everything. Man.
Yeah, yeah. I mean, you can just put a map in here and just.
Yeah, I saw what you did and still handwrite it.
Yeah, it's awesome. I love it. I mean, it's really nice, and it's not weird on your eyeballs either. You know, the screen, like some computer screens bother my eyes. This really doesn't. Yeah, this one is really.
Next time I get one.
You have to figure out the cheaper version.
That's good.
A cheaper version of a digital notebook. I can't afford an iPad.
But you know what? You might be able to, because there are student discounts that are totally cheap. And Harvard has to have like an even bigger discount than normal places.
Did you know that? I know if you're a student, you just go to an Apple store and they give you a discount.
Yeah, but each institution also has an added on top of that discount. Like I can shop as a faculty member, and it saves me like it's a lot. Yeah, and I think students. I know a couple of my grad students have bought computers, and it's like 40% discount.
Yeah. Maybe I should buy another computer before my break.
Yeah.
Before I leave.
We're not going there. Arch trap line.
Yeah, that's better. And we're off. Almost.
Yeah, if you want. So, it's nice then.
The only thing you have to not do is let small children around it.
Okay, this is coming to the control room now. We are T-minus 20 seconds till launch. Just getting confirmation from the various desks. We've got telemetry confirmed. Ground station radio confirmed. Check. Auxiliary systems. Extraterrestrial systems. Main thrusters. Oh, 1 sec, we're getting the green. Standing by. Launch pad. Payload. Ground system control. We're getting confirmation that main boosters are coming online. Just standing by.
Jason, you ready for us to pull this lander? Yeah, I'm just watching you go for it. Alright, we're going.
Pasadena, ground control check. Houston.
Check. Release.
And we have main booster ignition. And there she goes. No strings attached on her first mission up to the surface. Everything looks good. Attaining an altitude of 8 meters, 9 meters, 10. Secondary boosters have come online. Everything looks good. In a minute she'll do a roll, going to a degree of 26 degrees. Optimum X. And ladies and gentlemen, that is it. That was Sebastian's last ROV dive of expedition FK190211. It's been an absolute pleasure having you guys along with us for this exploration of Poseidon's Pagoda, Sonora Margin, and the other locations that we visited. Now, my colleague Veit has put up a link. Please check us out at schmidtocean.org. And we wish you all a pleasant time until we next go live, which will be in a few months. We're off now for the vessel to go into dry dock, refit, and Sebastian will also be going into a refit, and we'll be back out in, is it July? June, July. Until then, stay safe. This is Falco and ROV SuBastian signing off, and good night.